we're gonna, as Olga said, kind of move from doing just kind of static quantitative proteomics into some of these more dynamic systems. So I'm really excited for this set of talks. If you didn't see on Monday, some quick disclosures. I'm currently a postdoc researcher at the University of Pennsylvania, but I also do have a company that I consult for called Talis Bioscience. So these days lately, I've really been interested in this idea of nuclear integrity. So what does that really mean? The nucleus inside of our cells is the house for our DNA, our genome, but it's also organized and read and written by proteins. So here I have two examples of two systems. I have cancer and I have aging. And just visually, it's very striking. And you can tell immediately that between healthy and these different disease states, the nucleus becomes very different. So here you can see in leukemia, the nucleus is typically quite small and compact, but then under the disease state in cancer, the nucleus becomes large, irregular, disorganized. When we look in aging, young nuclei are kind of smooth and compact, and then in older cells, in older people, the nucleus loses some of that integrity. And as the nucleus loses its integrity, this is really a function of the proteins that are trying to keep the nuclear membrane intact and keep the chromatin organized, losing their structure and losing their function. Now, nuclear proteins are actually a little bit difficult to study. So in our central dogma, we have DNA genes, which then get read by proteins. This turns transcription on, and then we get RNA expression, and then the protein is finally translated. There's really great measurement systems for studying chromatin biology like this. And for studying DNA, we have next generation sequencing, which has come such a long way in the last 20 years or so. We can use sequencing technology to do things like RNA-seq to measure RNA molecules. But if we want to actually measure the proteins that are organizing, scaffolding, reading, and writing DNA inside the nuclei, we really need to use something that reads the proteins themselves. So I, of course, use mass spectrometry for doing this. Mass spectrometry is super powerful. Hopefully you're all here because you don't need to be convinced of that, because we can make so many comprehensive measurements of the proteome. We take all of these mass spectra, we turn them into chromatograms, and then we're able to make these molecular maps, these phenotypes of what's happening to the proteome under different subtype conditions, cellular subtype conditions. Now, this is using protein abundance as kind of this static number, right? So here we just have all these samples, we have all the proteins, and we have how much we measured of them. But proteomes are so much more than just a sum of their parts. So later today, we're going to be hearing more from Isa about things like protein-protein interactions, the interactome of proteins. It's not just a single protein acting by itself in a cell. Typically, it's got its friends, its social network that it's going to be affecting structure and function for. There's protein localization. A protein that's in the cytosol versus a protein that's in the nucleus is going to be doing very different functions. And commonly, these are proteins that are going to be regulated, controlled by post-translational modifications. There's a lot of different types of proteoforms, and here I'm talking mostly about bottom-up proteomics, but different proteoforms and measuring them can be very dependent on specific sites, specific amino acids that are being modified, say by ubiquitin, by phosphorylation, or very commonly these days, people are starting to study glycosylation. And then we like to think of protein abundance as this single number. It's a quantity, but it's really not a static number. It's a snapshot of a dynamic number that's changing between proteins being synthesized and then proteins being degraded. So all of these today, we're going to talk about measuring them using various forms and applications of mass spectrometry. So I'm going to start talking about making measurements in quantitative mass spectrometry to start. So you've heard a lot this week about DIA, and we've heard from Brian Searle on Monday, and we've heard from Brendan McLean the past couple days. That quantitative DIA, there's a lot of methods, and the method that Brian and I use most commonly is this idea where we have our set of samples, We mix them together so that we have this library, this pool. We acquire all of our data. In the middle of our single shot DIA, we acquire 6 gas phase fractionated runs of that pool. So it's just multiple injection of that one pool 6 times. We build a library, a chromatogram library, using that data, commonly using predicted spectra from PROSIT these days. And then we search our samples for this library. So this kind of breaks down these steps into asking a question about what can I detect, and then asking a question of what I can quantify. So I want to dig a little deeper into what I can detect versus what I can quantify. So here I have a distribution of the yeast proteome. So this is showing in the log 10 protein copies per cell, all of the proteins along their abundance density. So this would mean that proteins on the left-hand side of this figure are low abundant proteins. Proteins on the right-hand side of this figure are high abundant proteins, and then somewhere in between is all of the other proteins. And most proteins that don't have extreme abundances, they fall somewhere in the middle here. If we were to measure this ground truth yeast proteome with mass spectrometry, we cannot detect everything. The things that are easiest for us to detect with mass spectrometry are these higher abundant proteins. Because they're higher abundant, they give us stronger signal in the mass spectrometer. We're more likely to make a detection. It's harder to detect proteins that are lower abundant. If we then ask the question, so this is what I can detect, we might want to say everything I detect I can quantify, right? But that's not the case. When we look at the peptides and the proteins that we can quantify, we see the exact same trend again, a subset of what we can detect. So this is showing overlaid in green. These are all the things we can quantify now. And you can see, just like detection, we can quantify very abundant things easily. But as proteins get less and less abundant, it becomes harder and harder to not only detect them, but also to quantify them. This is important because a lot of times the stuff that's low abundant is really important biologically. So here I'm just pointing out 2 particular proteins that have a lot of interest biologically, a TOR target of rapamycin protein and a sirtuin, SIR2. You can see that these proteins, very, very important biologically, fall on the bottom end, on the bottom half of this abundance profile. So these are proteins super important, but maybe hard for us to see. So this kind of paints a picture, this detection versus quantitation picture, that mass spectrometry is not inherently quantitative. When we measure things with mass spectrometry, we kind of assume that higher quantity will give us a higher signal. Lower quantity will give us a lower signal. And that signal, typically when we're doing DIA mass spectrometry, we're using an MS2 chromatogram. So if we say this small quantity, small signal, we have a small peak area. When it's a higher quantity, a higher signal, we should have a larger peak area. But at some point, even though the quantity decreases, there is no change in signal. So when we plot these quantity versus signal type charts, we kind of get a hockey stick shape. This is because every single analytical instrument, not just mass spectrometers, have this limit of detection, this limit of quantitation. At some point, even if you decrease the quantity that you're trying to measure in the mass spectrometer, you no longer get a change in signal. That point at which you've decreased the quantity, but there is no change in signal, that's a limit of quantitation. Why is this limit of quantitation important? Every single mass spectrometer and every single sample prep and every single peptide has its own unique LOQ, its own unique limit of quantitation. The lower the limit of quantitation, the better the biomarker is. This is because it means that that analyte, that peptide, that protein, that thing you're measuring can be measured more sensitively. You can get to a lower and lower quantity correlation for the signal that you're measuring on your instrument. What exactly does that mean in a proteomic standpoint? Here I'm taking one protein from yeast. This is a very abundant protein. It's a membrane-bound protein, so I've just marked out in gray where the transmembrane domains are. Similar to next-gen sequencing, we can make kind of a read pile. So here, all of the black boxes that are on top of this protein sequence represent a different peptide that we've detected with DIA mass spectrometry. So now we have all of these boxes stacked up. These are all the things we detected. But if we look at their calibration curves, look at their limit of quantitation, they're all very different. So here's one peptide that has a lot of variation in its signal. This is a peptide, there is a correlation of signal to quantity, but it is highly variable. It's very difficult to say for sure that some signal corresponds to a change in quantity versus just variation on the instrument itself. Compare that with another peptide, again, from the exact same protein. This peptide has a much smaller variation in its signal, so it's much easier for us to make the call that, yes, this change in signal corresponds to a change in quantity. We can pick a third peptide, all from this same protein, and this peptide even has some overlap with this other peptide. We look at this peptide now, and the variation is even smaller. So now this particular peptide, the signal can very easily be tied to a change in quantity. So this means 3 peptides, all from the same protein. One of them is not really quantitative at all. Another one is a decent biomarker, but not the best. And then finally, we have a peptide that would be the best peptide proxy for this protein. Now, this is just an example of one protein and 3 peptides in this protein. You can imagine every single peptide has this value. And it depends on the type of proteome you're measuring, on the way you prepared your samples, and on the mass spectrometer that you're using. This is not a static number that is the same for every single peptide, no matter what you're doing with it. So this means that now every single protein, every single proteome that we're trying to measure is a little bit different. So what we can do is build these kind of calibration curves. We can build them by picking whatever proteome we're interested in. So if we're interested in cerebrospinal fluid, we can pick an example of reference cerebrospinal fluid. You could actually just buy these from companies like Golden West. And then you can ask the question, what can I detect in cerebrospinal fluid? So we did this experiment, and we found for us in this healthy donor pool of cerebrospinal fluid, we detected a little over 1,000 proteins in that healthy donor pool. So now I've just emphasized multiple times, not everything you can detect is quantitative. When we look at the calibration curves for all of those peptides we detected, we find that there are about 666 good biomarker candidates. So this is things that had any change in signal when we change the quantity using these calibration curves. Now, typically to do this step would take weeks to months, because every single one of those proteins that you're interested in, you would order stable isotope labeled standards for each one of the peptides you want to assess, and then build the calibration curve, measure the data, and analyze LODs and LOQs. So next, you might want to do the highest quality preclinical biomarkers out of this. So now you're doing more like calibration and validation tests that require those heavy isotope standards. And as I mentioned, every single one that you want to purchase is going to cost you about $1,000 per peptide if you're doing a heavy isotope standard for them. So this process using kind of conventional methods is going to take you weeks to months just to decide which biomarkers you want to chase after from your system. Then it's going to cost you about $1,000 per peptide that you want to test for preclinical validations. If we use DIA, now we can measure every single protein, all of the peptides in the sample. If we use this approach that I had published last year called matched matrix calibration curves, now instead of spending weeks to months to figure out what you can detect in your sample, it takes you 2 days. You do one of those gas phase fractionated libraries, you figure out what you can detect in your sample, and then instead of spending $1,000 per peptide for stable isotope labeled paired standards, you spend about $1,000 per proteome using either SILAC to shift the mass MZ values of your peptides, or you can use other methods to dilute your reference proteome in a background. So DIA has already really enabled a lot of advances in doing quantifications and doing them very quickly. So I'm going to change gears in just a minute, but I could also take some questions. I see there are already some questions in the chat.
Yeah, there's one question which may be relevant now. So from the proteomics point of view, could you please kindly summarize what is a good biomarker?
Yeah, so proteomics biomarker, there's a couple good papers, and if one isn't able to get to them in the chat, the two good papers as far as biomarker validations, the first one is Andy Hoofnagel and Russ Grant. The title of it is something like Paradise Found or Paradise Lost and Found. It's about a 3-page paper, very quick read, that highlights all of the experiments you should do to get to this kind of preclinical biomarker stage. Again, that's not like a validated assay to put in patients, but it gets you to the preclinical stage. The other paper, senior author, last author is Steve Carr, and it's the Fit for Purpose paper. So these two papers are kind of, I would say, like the gold standards for doing preclinical biomarker discovery in proteomics. There's further clinical validations. CLSI has a workbook, but that's like super thick of all like kind of the FDA regulations to get to an actual clinical biomarker. So it's a great question. I think we got that one. Question from Ahmed, how did we determine the LOQs of peptides? That's in this paper, which we're not going to like dig super into the details, but this J Proteome research paper from last year goes over the methodology for building the actual samples for calibration curves, and then it also has link to the data analysis to calculate LOQs from your data. You can also calculate LOQs using MS Stats. I'm not sure if you're going to learn that later this week, but once you learn how to use MS Stats, it's very easy to use this function in MS Stats to calculate LOQs. I'll start at the top. Raja has a question. Protein copies per cell is being done using histone proteomic ruler and DDA LFQ. So the protein copies per cell that I had on those first slides, that's taken from a paper by Sina Gamagami. Great proteomics work, but this was like a Herculean effort in yeast where every single ORF, every single open reading frame in yeast was tagged, and they did like actual counting to get those protein copies per cell. So we use that as the ruler in that case. Is there a good agreement in copies per cell between DDA and DIA? I was not going to talk at all about comparing DDA and DIA, but there are some great papers. I'll see if I can dig them out that are comparing that. A little bit later, we're going to do some comparisons of DDA versus DIA quantification. DDA is good for like a limited dynamic range, but DIA has a much broader dynamic range. So if each individual peptide has its own variation, is this taken into account during quantification in the case of shotgun proteomics? As far as I am aware, there is no algorithm out there that really takes into account that different peptides have different LOQs. I think most protein quantification, you'll be learning more about this later too, I think most protein quantification, I think the most common way people do it is literally just summing peptides to get a protein level quant. Like a top 1 or a top 3 or a top n, just summing everything up or taking the average. There are some more intelligent ways of doing this that MS Stats takes into consideration, but that does not include like specific LOQ values. Coco, we will post names of papers. If the TAs don't get to it, remind me in the chat, and I'll get links for later. And these slides will be up after the talk today. So you'll have the DOIs for that. Okay, so I'm going to move on for now. I think we got mostly everything. And maybe I might get back to some of these questions later. Yeah, so in the context of biology, I had mentioned earlier how now we have these giant maps of looking at differential abundance as a molecular phenotype for different perturbations, different molecular subtypes. So one question that I had asked previously was whether there was a different molecular phenotype for lifespan extension. So here I was using yeast, so everything was being done in this yeast model organism system. But what I wanted to ask is, can we extend lifespan? Can we have yeast that live longer, either through genetic mutations or through environmental interventions? And I kind of expected I was going to see this phenotype reflected in protein abundance. And that's because there had been previously this paper where they had measured yeast lifespan by deleting every single gene in yeast. So another Herculean effort, they deleted every single gene in yeast systematically and measured the lifespan of the yeast. And they found that there were some pathways that extended lifespan. These yeast lived longer. There were kind of major themes here when they deleted each of these single genes, which deletes the protein. And they found that a couple of these pathways were things that to me really spoke to the protein level and the proteome itself. Things like ribosomes, so that spoke to me saying the synthesis of proteins is being affected. The proteasome, the degradation of proteins is being affected. SAGA is a protein complex in the nucleus that kind of can regulate different gene expression. So kind of all of these taken together, I was thinking there must be something in the proteome then that is driving this phenotype of extended lifespan. Let's look at protein abundance and see if we can find it. So I picked a couple of these single gene deletion yeast strains and I wanted to grow them up in bulk, measure their proteomes with DIA, and I was going to see this beautiful signature in the proteome. I did what's called a set cover approach, and what this set cover does is, given two sets of information, I gave it the functional overrepresentations, and I gave it this protein interaction network because I wanted to optimize for a couple things. I wanted to optimize for single gene deletions that extended lifespan across a range of values. So I didn't want all very large lifespan extensions. I wanted to get a range of lifespan extension. And I wanted to get a number of unique genetic interactions. So I didn't want to do all gene deletions that were ribosome gene deletions. I didn't want to do all that were minosyl-transferase deletions. I wanted to kind of cover as much as possible across the functional overrepresentations and have the most unique genetic interactors, the most unique protein-protein interactions. I ended up with this list, which surprisingly included two of the SAGA complex members, which was interesting in itself, but seemed to span the range. None of them were slow growing. They had a lot of different genetic interactors. So I thought for sure I'm going to see like massive changes in the proteome when I compare these single gene deletions to regular yeast. You can probably tell by my slightly sarcastic tone, that's not at all what we saw. So when we delete TOR out of yeast and measure the change in protein abundances between that TOR deleted yeast and the normal parental genotype yeast, really not a lot is changing here. A couple of these gene deletions are actually just artifacts in the genetic makeup of the yeast itself when they made these single gene deletion strains. I look at a couple of the others, and again, I really am not seeing huge changes in the proteome. The biggest change that I ever saw was this SGF73. So this was really not promising for my hypothesis of this molecular phenotype of lifespan extension in yeast when I looked at each of the single gene deletions. So it was not surprising when I tried to say, is there a proteomic signature of lifespan extension in yeast? There really was not. There's really nothing here. Even when I compare lifespan shortening, so shorter lifespan yeast, there's really nothing in the proteome here. So this was really surprising to me because I kind of thought, like, obviously this has to be a change in the proteome. There has to be a proteomic signature, because what other molecule is going to be affecting the phenotype? It's not going to be the DNA that just kind of sits there. It's not going to be RNA. RNA doesn't really do anything. It's got to be the proteins, right? Didn't see that, at least in the abundances. So I kind of put that disappointing result to the side and I said, okay, it's not single gene deletions. What about environmental interventions? So it turns out actually you can put yeast on a diet just like a person, just by changing how much glucose it has available. So at these higher glucose levels, calories are abundant, and the yeast has a phenotypically normal lifespan. At some point, when you decrease the glucose too far, when you limit its calories too much, the lifespan goes down, kind of starvation type of thing. There's a sweet spot in between, however, where the glucose is present but not overly abundant, where lifespan extends. There is a point of calorie restriction where lifespan increases on yeast. And I said, okay, this is going to be it. This is where I'm going to see my proteome changes. And I kind of hypothesized across a range of glucose availability, I might see that reflected in the proteome a couple different ways. So the first would be across availability, the peptide abundance, the protein abundance would not change at all. These are proteins that are not affected at all by the amount of glucose. I might see a dose-dependent change in protein abundance, where maybe a particular protein is highly abundant when glucose is available, and then lowly abundant when there isn't glucose, or vice versa, low abundant in high glucose, and then becomes high abundant when we move to low glucose. When I looked at my data, I didn't really see either of those trends. So each one of the lines here represents another peptide, and I'm plotting those lines across the different glucose availabilities. So I didn't really see either of those trends. Mostly it kind of looks like flat lines. But one thing that I did notice when we were looking at this was some proteins seem to almost have a signature where they're high expressed in high glucose and then low expressed in low glucose, kind of like a switch. So it's abundant at the same level in one condition and then abundant at a different level under a different condition. So when I reanalyze my data that way, where I group the high glucose and then the low glucose and compare those proteomes against each other, I see a huge change in the proteome abundances. This was more what I expected to see when I was deleting genes out of yeast. So comparing the low glucose to the high glucose, there's a lot of protein changes here. And this is something you can do. I just made these figures directly in Skyline using the group comparisons. So if it looks familiar, that's why. When I look at some of these functional groups that are being enriched or depleted between the glucose conditions, it kind of makes sense. So under limited glucose, things like the ribosome biogenesis are being downregulated. Under low glucose, things like carbohydrate metabolism are being up-regulated, increased abundance. So these kind of make sense in the context of the glucose level, but didn't make sense in the context of lifespan extension in yeast. So some of these proteins that we did see with increased abundance in that low glucose sample are documented, known as having lifespan extension properties under calorie restriction conditions. So this is one protein, a heat shot protein, HSP12, and I'm showing the peptide trace from in Skyline, where you can see for this protein, Skyline plots each one of the peptides for that protein that is measured for that protein. So each line is another peptide from this protein, and then each one of the points along the horizontal axis is another one of those samples. So you can see this protein, all of the peptides are low, low, low in these first three glucose conditions, and then all of the peptides very nicely, stably become increased abundance once we hit somewhere between 0.5% glucose and 0.05% glucose. At some point in there, this gene is turned on, transcription is turned on, and the protein becomes abundant. So that was kind of interesting. It definitely didn't prove my hypothesis right at all. My hypothesis was definitely proven wrong. And I kind of took away from this 3 lessons. So the first lesson I learned was to my surprise, just deleting a gene might affect a phenotype, but it didn't seem to affect the protein abundance. The proteome seemed pretty plastic, pretty stable in response to deleting an entire gene, which was not what I expected. I found that although genotype manipulations didn't seem to change the proteome, calorie restriction really did. So environmental modulations had a much stronger effect on the proteome. And then finally, what I really took away from this is that protein abundance alone doesn't necessarily mean there's a mechanism of action. So there has to be a way to get beyond the protein abundance and ask questions about the functional proteomics. So you might tell we're getting ready to switch gears. So I'll stop again for questions, but it looks like the TIAs have done a pretty good job. There was a question about the low glucose conditions. They were exposed to low glucose for roughly 24 hours. I tried to control for growth rate and harvest so that the population doublings were about the same for each. So the time was a little different for each of them. Yeah. So we arrive at this message: proteomes are more than the sum of their parts. Protein abundance is great, but isn't always the full story. Sometimes you see no changes in the proteome, but there's clearly something happening molecularly in these cells. So I had kind of alluded to this, but the next logical place to start looking, to start exploring, is in protein turnover. So I mentioned how protein abundance is thought and represented as this static number, but it's actually a very dynamic value that we're only taking a snapshot of. So the protein abundance will increase if you're synthesizing more protein or if you're degrading it less. And the abundance will decrease if you're degrading it more or synthesizing it less. And what we see when we're measuring a protein abundance is just at that moment in time, the snapshot of these two molecular processes. So using mass spectrometry, using this technique, pulseSILAC, can capture both of these pieces of information. So in a pulseSILAC experiment, you would take your cell culture. For me, I was working in HFF1 mammalian cell line, and you grow your cells in regular media to start. Once the cells are happy and proliferating, you switch the cells to a heavy media. So this is media that you create where you purchase heavy amino acids. You purchase media that is depleted for those amino acids, and then you supplement your media with these heavy isotope labeled amino acids, typically lysine and arginine. So now, as time passes, as your cells are growing, as they're synthesizing proteins, you are the cells are including these heavy isotope labeled amino acids to their proteomes. So if you look at a ratio of the light to heavy proteins in these cells at time 0, where they've only seen light media, all of their proteins contain 100% asterisk. It's not entirely 100%. It's naturally abundant isotopes, but 100% light amino acids in their proteomes. And then as time goes by, they start incorporating more and more, synthesizing more and more heavy proteins that contain these heavy amino acids. At this point, this 50-50 point, it's called the protein half-life, so the turnover value. So when I look at this as a quantitative proteomicist, I notice two things. The first is that all of these measurements start at 0. So this curve here, to get to the 50%, to shape this curve, having these initial points is very important. Having these low abundance ratios is very important. The next thing I notice is that these protein synthesis values occur on the order of hours to days. Some proteins are extremely quick and are turned over very quickly. Some proteins are very stable and stick around for days. So these are kind of two aspects that suggest we need a very sensitive ability to measure dynamic range. So we need dynamic range in the time dimension. We need dynamic range in this ratio measurement dimension.
Lindsay, there's a technical question. Are these time scales, for example, in terms of hours, typical of protein turnover?
Are these time scales typical of protein turnover? Yes. Yeah. So these time scales between like hours to days is typical of all kind of protein expression. Every protein is a little different. And honestly, every proteoform is a little different. So we'll talk a little bit about getting at that proteoform question in just a bit.
And another
Yes.
question related to this. So 50-50 light and heavy, shouldn't the ratio of light versus heavy be 1?
Yeah, sorry, this label that I gave it here should be over total. Sorry about that. That was my fault. I'll definitely fix that slide before I post it. Have I seen protein turnover in 30 minutes? So when you model these, it really depends whether you see turnover in 30 minutes will really depend on how many time points you collect really quickly there. There is a full range is what I will say. Susanna question, will the curve be slightly different for every protein? Absolutely. Every protein will have a slightly different curve here. It's a protein dependent process. I will let one of the TAs grab Ahmed's question because that is something I think that they can answer. And I think maybe BK's question can be answered by the TAs too. So I'll keep going. So I think kind of the logical thing that everyone's thinking, considering this is a DIA focused workshop, the question becomes naturally, can we do pulseSILAC and measure it with DIA? One of the first questions is, how do you analyze that? How do you process this data? So I just described kind of this protocol of how you would start cells in a light media and then grow them into a heavy media. For these experiments, I did the exact same style of gas phase fractionated DIA library, the chromatogram library approach that I use in my other experiments. Here, after you're done collecting your data, you have to do a bunch of post-processing in order to calculate these half-life values. But kind of this general process, you do your pulseSILAC experiment, you acquire your data, and then you calculate these protein half-lives. Just to give a little bit more detail on this vague acquired DIA MS data, what we thought to do is for this chromatogram library, because we're doing pulseSILAC, we're really only interested in the longitudinal changes in abundance after time 0. So what we can do is we can generate a time 0 chromatogram library. And just like Brian had talked about, I like to use PROCIT to predict my spectral library. And then I use DIA analysis and gas phase fractionated DIA analysis using Encyclopedia. And now I've built this time 0 library. Then using that time 0 library, again in Encyclopedia, I detect all the peptides in all of my samples throughout the pulseSILAC time course. And what that does is it performs a retention time alignment for me. So now I have all of the peptides that are detected and the retention times aligned. And now I can use Skyline to pull in those chromatograms for the light and the heavy paired proteins. This is very quick to do in Skyline. You guys are all becoming Skyline experts now. But what we can do, we just add all of the peptides to our target list using that library from Encyclopedia. And then you can very quickly, in about 3 clicks, add the heavy isotope labeled standard version of all of those peptides. So now I have this workflow using all this free open source software that you've heard about this week. The question becomes, how does it compare to DDA? So we wanted to benchmark this. Sounds like a great idea. In theory, maybe it'll work. We're not sure. So we took a heavy proteome and a light proteome, and we mixed them together in ratios. This is kind of from the Navarro paper some years ago. This is kind of the idea of that hybrid proteome. So now we have known ratios that we can compare to our measured ratios. So if we do a conventional pulseSILAC, this is pulseSILAC-DIA, we see that the measured ratios, those are each one of the colored points, the measured ratios, and we can compare them to the expected ratio. That's these horizontal lines. The N up here just indicates how many detections that we made. So out of all of those detections, we can visualize this with a box plot to compare the measured ratio and the actual ratio, the expected ratio. We can see for some of these less extreme ratios, DDA, conventional SILAC, does a pretty good job of recapturing the expected ratio. So you can tell that these box plots are kind of small, and the mean is right about at that expected ratio line. But once we get down to about a 1% ratio, so that's 1 to 99, the measured ratio has started to kind of already hit its floor. So we kind of get this leveling out of the measured ratio once we get down below this value. We can compare the exact same samples measured with DIA using that pulseSILAC approach. And one of the first things that we spot visually is that the abundance range, the dynamic range of the measured abundance is much wider when we use DIA. DIA is being done on the MS2 level. So we get a wider dynamic range in a measured abundances, but we also gain an order of magnitude in quantitative accuracy. So that order of magnitude is this light blue box is now jumped down more closely to the expected 1% ratio than if we can compare the conventional pulseSILAC samples. So we wanted to apply this in maybe a more interesting system than this benchmark. And what we decided to do was look at bortezomib treatment. So this experiment is performed the exact same way. So we're taking these human cells, growing them in light for a while. Then we're going to switch them to heavy media. But at the point when we switch them to heavy media, we're going to treat one set of samples with DMSO. The other set of samples we're going to treat with this drug, bortezomib. So bortezomib is a proteasome inhibitor. It's the first FDA-approved, first-in-class proteasome inhibitor used primarily for myeloma indications. This proteasome inhibitor, what we expect is proteins that normally would have been degraded by the ubiquitin-proteasome pathway are now no longer going to be degraded. So they're going to be around longer. They're going to have longer half-lives, longer turnover values.
Lindsay, a question related to that. Why do we use DMSO as a control in this type of studies? Is it truly inert?
Yeah,
Why not just add a smaller amount of molecule water?
Yeah, that's like an excellent med chem question. DMSO is just kind of the standard. I actually don't know historically why we don't use water, but definitely in med chem, everybody uses DMSO for their control. Yeah, usually Leah has a good point in the chat. Usually when you're bringing up these drugs, you get the drugs as like a lyophilized like powder, and typically you resuspend them in DMSO, not in water. Yeah, but I'm not sure why we, like science as a field, originally picked DMSO to reconstitute these versus something else.
Yeah, another comment by Raja. Yeah,
sorry, just to mention, I think it's because DMSO can actually dissolve way more things that you could in just an aqueous solution.
Yeah, there's definitely the argument for like using water is only going to get water soluble things, and some drugs are not easily water soluble. So yeah, I guess while I'm looking at the chat, I do not personally have a good resource for examples of protein turnover timescales, but if someone knows one off the top of their head, please let us know and we can hook that up. How does pulseSILAC-DIA compare to pulseSILAC-TMT in terms of accuracy and precision? I have not seen a paper comparing that with pulseSILAC, but Professor Vitek, Olga, actually has a good paper with, I think it's Lucas Reiter, that compares DIA with DDA. Maybe someone can drop a paper for that. I don't know it off the top of my head.
I will add the reference in the Google Doc.
Perfect. Yeah, but that's an excellent paper. Kind of one of the take-home messages I got from that paper comparing DIA with TMT is that they are about the same in terms of accuracy and precision. And it's kind of just whichever one you're more comfortable doing. Like, for example, if I tried to compare DIA and TMT, TMT would probably look awful just because I've never done it before, but I've done DIA a lot. I'm sure there's other labs out there that are very, very comfortable doing TMT, where their DIA might not look as good as their TMT.
That's pretty much the general conclusion.
Perfect. Glad my too long don't read version of the paper hits the right take-home message. So I haven't seen it in pulseSILAC. There are additional confounders with pulseSILAC that maybe that would change. Just kind of like the isotope distribution of the SILAC on top of the isotope distribution, or I guess not isotope because that's MS3, but the TMT takes advantage of isotopes. The pulseSILAC takes advantage of isotopes. It might work out because it's kind of different isotopes, but at the same time, it might cause some interferences. It's like a really good question. Good thought experiment. You could probably do some good in silico simulations to see if that would work. Okay, I think we're caught up with questions. I'll just share with you some of these results. This is what those turnover curves look like. So this is showing the time as it progressed along the horizontal axis, and then on the vertical axis, it's the fraction of the light to the total protein abundance. So when we start out at time 0, all of the proteins are 100% light. But you can see even just by eye looking at these curves that some proteins really are not being turned over at all. They remain 100% light pretty much forever. So those are proteins that are just not being synthesized more, and the light is not really being degraded. The light at time point 0 accounts for pretty much all the protein that's there forever. And we know this is the case, right? Because like there's proteins in your eyes that you're born with that you never get new proteins in your eyes. That's it. So we know that this is the case for some proteins. You can see the majority of proteins turn over at like kind of a moderate rate, about maybe like 150 to 100 hours. But we can calculate these actual values by calculating the 50% point, the half-life in these curves. And when we do that, we can make a pseudo volcano plot out of the differential proteins. So this is not a real volcano plot. So I'm going to kind of explain it a little bit. So here, instead of a log2 fold change, we put the log2 of the turnover value for bortezomib over the DMSO. So if there's not really any change, it's like a volcano plot, not really going to be interesting. The larger the log2 value of this ratio gets, the more different the turnovers are. And then we get these p-values. So if we look at the statistically significant differences in turnover, we get this handful of proteins that are being differentially turned over under bortezomib treatment. If we look up some of these proteins, they cover a range of protein half-life values. Real quick, I'm going to point out in red, these 3 proteins are known. They're documented as being part of the ubiquitin-proteasome pathway. So that's reassuring that we're recapturing some things that we know about the ubiquitin-proteasome pathway. One thing that was interesting to me is these 3 proteins that actually have an opposite half-life. They're not being extended, they're being turned over faster, it seems. So these are kind of interesting things to see. It kind of makes me wonder, you know, this decrease in the cathepsin D, this CAT-D protein, the second one here, could it be explained by, one, it could be an increased synthesis rate. Cathepsin D is an aspartyl protease, so it's kind of maybe orthogonal to the ubiquitin proteasome pathway as a protease itself. Maybe cells are trying to upregulate this protease activity to try to clear out some of these accumulating proteins that aren't being degraded normally. Cathepsin D, this is also a precursor. Is this a proteoform that we're not capturing because we're just doing kind of regular bottom-up quantitative proteomics? So could this precursor just have another form, another isoform that we're not really capturing well here? So this idea of even when we break down protein abundance into its component synthesis and degradation rates, there are still things functionally that we can't quite explain at the proteoform, at the functional level, kind of led me to some of my next work. So it looks like we might be doing okay for questions. So I'll keep going real quick. So I mentioned in this last work, looking at protein turnover, we were looking at bortezomib, which inhibits the proteasome and the ubiquitin-proteasome pathway. So logically, that kind of led to my next interest, which was looking at post-translational modifications, ubiquitin. So in a lot of my systems, what I'm interested in is processes like aging or like cancer, where the DNA itself might not change at all, but the proteome, what's driving phenotypic differences. And in my postdoc at the Garcia lab, it's not too surprising, the main PTM we're interested in is histone tails. So in the context of aging, as you age, there's a lot of changes going on in your nucleus. So inside the nucleus, under normal chromatin states, as we age, we start to accumulate different protein variants in our chromatin. There's certain histone proteins that become incorporated more and more with your DNA as you age, as cells age. There's different histone modification patterns. The DNA itself might become mutated, and the DNA itself might be modified differentially. So measuring histones themselves, there's really only like the 4 canonical histone proteins that make up a nucleosome. But when you compare the complexity of those 4 proteins and all their possible PTMs, the problem becomes very large. For context, out of like the 40,000 human proteins, there's about 1E8 peptides to search for the proteome. There's only 4 histone proteins, but when you take into account all of the combinatoric PTMs that are possible on them, this becomes as hard a problem as comparing the size of an Egyptian pyramid to the size of the entire Earth. So it's a very difficult problem. So since we're kind of talking in the context of Skyline, I want to highlight some of the difficulties, not just in this search space, but also in Skyline, even when you're looking at these proteins. So here's one protein. This is histone 4. It is a mess. It is an absolute mess. So histone 4 can have a number of different residues: lysine 5, lysine 8, lysine 12, and lysine 16. These 4 residues, any one of them can be acetylated. So this is an example of that combinatoric problem. So each one of those residues, one of them could be acetylated. The others can be unmodified. 2 of them can be acetylated, or 3 of them can be acetylated, and then finally all 4 of them can be acetylated. So this is one peptide from one protein and just one type of PTM. There are other types of PTMs that happen on histones. So not only is that difficult just from a search space problem, but I'm showing you their chromatograms, and I'm showing you that for each type of modification, whether it's a single acetyl, double acetyl, or a triple acetyl, chromatographically, they do not resolve. So now we get all of these peptides co-eluting on top of each other, and they all have the exact same mass to charge value because they all have the exact same sequence and the same number of modifications. So their precursor mass to charge is the exact same. So this is just one example of one peptide on one protein that makes histones so tough. There is also, I mentioned how when we age, there are different variants of proteins, of histone proteins being incorporated into our chromatin. So here I'm comparing histone H3.1, so this is one histone H3 variant, with histone H3.3, another variant. There's a single amino acid change, an alanine to a serine. So when we look at this peptide sequence in one protein versus another protein, there are 3 peaks here. Which one is correct? There's a slight mass to charge shift, but which one of these peaks is the right one? If we're using DIA, unless we have a large enough MZ shift that it falls in a different DIA window, a different DIA isolation window, we are co-fragmenting these. So which one of these peaks corresponds to the right peptide? So histones, all of this to say, are very, very difficult to analyze. Some of the work that we've done is using Skyline and using this idea of the prior information, using things like spectral libraries, using things like IRT calculators to process our data and look at our data, so that we can be more convinced that what we're measuring and what we're reporting is correct. Oh, there was a quick question in here about derivatizing the lysines. All of this that I showed you, this is all with the lysines derivatized. This I say it's unmodified, but it's actually propionylated. So these are all derivatized, and chromatographically that does not change their elution off of C18 column at all, which is unfortunate. Okay. So we're going to use Skyline. We're going to use as many tools as possible in Skyline to make our life a little bit easier. So I put together kind of this collection of scripts. They're definitely still in a work in progress stage. But what this collection of script is going to do is first, it's going to help you build a Skyline document. So this is especially useful if you're doing some of these histone mutants. There's also oncogenic histone mutations, point mutations in histones. This allows you in Skyline, you've seen how easy it is to add any new target you want. You can simply type in a sequence you're interested in. You can add new modifications. So commonly for histone PTMs, people are interested in kind of the usual suspects, things like methylation, acetylation, but there's growing interest in other types of modifications like glycosylation, like citrullination. Using Skyline, it's very easy to add modifications just by right-clicking on the target and going down to that modifications option in the drop-down menu. And then finally, doing non-model organisms. So I have collaborators that are honestly doing all kinds of organisms, from slime mold to leafcutter ants to, of course, human. So all of these are going to require a slightly different approach. Using Skyline, it's very easy to accommodate any of these experimental systems. Next, we're going to use DIA to acquire this data. And I'll talk a little bit about how we have to think about modifying a DIA method for different types of systems. Brian touched on this a little bit when he mentioned how if you're doing phosphoproteome and you want to do DIA, adding that PTM changes a little bit how you should set up your method settings on your instrument. And then finally, a lot of these analyses are going to be done using Skyline, but accompanied by some Python scripts that I've been working on just to make this a little bit easier. So the very first thing is just building that target list in Skyline. So the target list in Skyline on the right, just a snapshot here. We've all seen how you can add peptides and proteins, but one of the first scripts that I wrote was to take a list of modifications that you're interested in. So this has lysine 4 acetyl, lysine 9 trimethyl, serine 150 phospho. Take a FASTA of the proteins you're interested in, and then using what's called a Cartesian product, calculate all the possible combinatorics for those peptides. So it's important in Skyline that we're kind of monitoring for all these combinatorics to be sure that what we're seeing, the peaks, the chromatograms that we're seeing, are truly belong to that particular isoform, that particular PTM peptide. So now we have a target list. One of the other tools that we're using in Skyline is the IRT calculator. So when we prep histones, first we isolate histone proteins from the nucleus. But that isolation is not perfect. When we're enriching for histones, a lot of times we get other proteins that are co-enriched with those nucleosomes. We can use those co-enriched peptides to build an IRT calculator. So we don't have to use Biognosis. We don't have to use commercial kits to spike into our samples. We can make our own calculator from common contaminants in the sample, or here a lot of these peptides are ribosomes, not things we're interested in from an experimental standpoint, but things that we're going to be measuring anyway. And just as a reference in here, I'm not going to walk through this entirely, but building these calculators is very straightforward. I'll add this as a reference so you can kind of play with it yourself. But just when you get to this edit IRT calculator, in the drop-down menu, you can choose add, and then you can paste whatever peptides you want to use in here. There are some tips and tricks that we can talk about if people are curious to pick peptides that are going to work well. But the sky's the limit. Anything that's in your sample, you can build an IRT calculator for. Okay, so we're built. The Skyline document is ready. The next step is to acquire data. So acquiring the data in DIA, we've now looked at DIA data a lot of different ways. This is going to be yet another way to look at DIA data. So here I have the chromatography on the horizontal axis. So this is the measured retention time for the histone PTMs in kind of like a standard PTM analysis. Then on the vertical axis, I have the precursor mass to charge of these histone PTMs, and I am binning them by the isolation window scheme. So using a previous method in the lab, we used 50 MZ wide windows, 50 mass to charge wide windows. So as we look at this one slice of retention time, some of these windows have 0 precursors. The next window up, 350 to 400 MZ, has 1 precursor in it. If we move farther up, some of these windows have 3 precursors in them. These are 3 peptides, 3 precursor ions are being co-fragmented in the window. If we expand that out, this is what that whole data set looks like using a 2015 method published from the lab. So this is not maybe an ideal DIA method using some of the things that we've kind of mentioned along the way so far today. So an ideal method for PTMs would not have co-fragmented isomers. It would have a fast cycle time. You would get enough of those points across the peak that Brian talked about. And then finally, it would be nice if we didn't have idle windows. We're not spending time collecting data in windows where there's nothing we're interested in. So a couple of things that we can do to optimize this method. We went from a 16 MZ, 16 windows of 50 MZ precursor range to a 25 windows of 24 MZ using that staggered approach that Brian mentioned earlier. So now we have 12 MZ effective windows. A couple things in this DIA method optimization. The first is we reduce the precursor range. Instead of acquiring the 900 to 1100 MZ range, up here, a lot of these peptides, a lot of these precursors are actually just lower charge states of things that are down in this busier section of the method. So we'll cut out these lower charge states and focus on these higher charge states that are down in the range of 300 to 900. The next thing we did by decreasing those windows and using that staggered approach, as Brian had mentioned, we kind of get twice the speed for nothing. We're getting reduced window size using these staggered methods. So we went from 50 MZ wide windows down to 12 MZ wide windows. And what does this mean? So let's take a look at just zooming in on a couple parts of the gradient. So this part of the gradient at this particular retention time in these 2 windows, we are co-fragmenting 4 precursors because these windows are so wide. When we decrease the size of the window, now we have separated 1 of those precursors from the 3. So we didn't do perfect. There are still 3 precursors falling in some of these windows, and those 3 precursors are within 12 MZ of each other. We can see other points of the gradient where we had 2 precursors that co-elute at the same mass to charge value, co-fragmenting in this window, but they don't have the exact same precursor value. So if we lower the size of the window, we decrease the size of the precursor window. Now we've separated out these 2 co-eluting isomers. Because they co-elute, but they fall in different windows, now we can more easily pick them apart in Skyline. So I'm seeing a lot of questions about what kind of MS. Here's a reference chart for you, and these slides will be up. So here's some proposed methodologies that you can use, just compared to kind of what the previous standard was. The 3 in the middle are Orbitrap instruments. And this last one is from a paper from the Dannon's lab doing swath, so on a triple TOF. I haven't personally been on a triple TOF yet. Someday I'd love to get on it, but these slides will be up later if you wanted to play around with this, these data acquisitions. But the last thing I'm going to talk about, analyzing the data. So now we've built our Skyline document, we've acquired our data, we've pulled it into Skyline. It's still not over. now it's the hard part. That was the easy part. So now it's the hard part. Why is it hard? I talked about these co-eluting, co-fragmented isomers. So here's another histone protein. This is histone H2. Lysine 4 and lysine 7 can be acetylated. Monoacetylated. The rest of these, the question before about the derivatization, the rest of these lysines are derivatized, are propionylated. So these two precursors, these two peptides, pretty much coelute. When we look at the fragment level, many of their fragments in the fragment letter are going to be shared. So in the Y series of ions, all of these ions down here are all going to be the exact same between these two peptides. What we should do to perform good quantification, to be confident in our quantification, is only use the fragments that are unique to each of these peptides, site localizing. In PTM, we talk a lot about site localizing fragments. So let's only include fragments that include the PTM modified residue. So when we do that in Skyline, we can mark these specific transitions, which are unique. And what that looks like is the dotted lines are not unique. Those are transitions that are shared between these two peptides. And then the solid lines are the ones that Skyline is going to use for quantification of peak areas. It'll still use the other fragments to perform detection, like peak detection, but it'll only use the solid lines for quantification. And that's very straightforward to do in Skyline. You can either right-click on the target itself, or you can open up the document grid, go down to the transition report, and then it'll show you all the transitions in here, and it'll give you a checkbox to say if they're quantitative or not. And I can link you guys to some tutorials that are still work in progress. They're not published or anything yet. You can very quickly, just in Excel, concatenate the precursor to the product MZ, and it'll give you a true false for whether or not it's quantitative. So now we can have this document, and you'll see that we can have in our document grid, we can check the things that are quantitative. We can look now at the precursor. The precursor is not quantitative because there's a lot of isoforms for this particular peptide. And when we look at the different fragment ions, we could see some of the fragment ions that are quantitative versus not quantitative. And then Skyline will not use the ions that are marked not quantitative. There's a lot of other helper scripts out there that you can look at. So this is that paper from the Dannon's lab that I mentioned before. But everything up until this point assumes that you're not doing anything else. You're only doing DIA for your data acquisition, and then you're going to analyze it in a targeted, like PRM style manner. If you wanted to discover new histone PTMs, you could do this kind of approach where you're pairing DDA with your DIA. And then you build a spectral library using whatever database search is your favorite. There's a couple that are better for histone PTMs specifically. But whatever database you like using, you can build your spectral library in Skyline using that bibliospec built-in tool. And then once you have your DDA library set up in Skyline, you have all the MS IDs in your document to pull in the extracted ion chromatograms of your DIA data using the detections in that DDA. So that ends up looking something like this. So when you have one of these paired DDA spectral libraries in Skyline, you can visualize each one of the MS IDs in that DDA search on top of your chromatogram, and it makes these vertical cyan colored lines. So you can see this chromatogram that has been detected in Skyline. It's picking the peak right here where we've got all of those paired DDA MS IDs. So this is a really powerful approach. We just spent so much time talking about not using DDA for our DIA analyses, but I think PTM analysis, especially complex PTM analysis, is a great place to go back to those spectral libraries and collect a paired DDA spectral library. It just makes analyzing this data so much easier because you've got like this huge blue highlight on where the peak should be. So as I mentioned, using kind of these tools versus some of the other histone PTM tools that are available out there has really enabled my collaborators to do a lot more with their epigenetic analyses. So I have a collaborator who's been working on epigenetic mechanisms of neuroblastoma. So one of the drugs that you use to treat neuroblastoma is this drug lorlatinib. It's an alpha kinase inhibitor, but some patients sadly relapse after getting lorlatinib treatment because they become lorlatinib, their tumors become lorlatinib resistant. So this type of mechanism, the collaborators suspect is an epigenetically driven mechanism where the tumor acquires this lorlatinib resistance. So now we can really quickly analyze this data. Now that we've got our Skyline document built, we've got this method to kind of plug and play analyze their data. We just pull that into the Skyline document. We can build, even for their specific system, an index retention time calculator specific to their preps in their system. I have another collaborator who was working in slime mold. So this is just a video that they have of the slime mold kind of like doing its slime mold thing. It's kind of cool and gross. But the slime mold goes from unicellular to multicellular. So that's obviously an epigenetic mechanism, right? So slime mold is obviously a non-model organism. And we're able to really quickly look at some of these different life stages in slime mold and assess the histone PTMs, the epigenetic marks, as slime mold goes from a unicellular to a multicellular organism. And it wasn't a huge surprise. The chromatin is more closed when it's unicellular and then more open in multicellular. And then I have a third collaborator who's doing epigenetics of ant socialization. So these are leafcutter ants, and they are clonal. So all the ants of a given ant colony share the exact same DNA, but you can see just phenotypically the size is totally different. Their roles are very different. And what he's been doing, he's been asking in leafcutter ants, yet another non-model organism, what is the difference in chromatin marks between these different casts of ants? So this has been a really fun project to kind of push some of the limits of doing good quantification in histone PTMs and PTM work that has really been enabled using all of the tools in Skyline and even coming up with some kind of hacks for how we can use Skyline tools in a slightly different way than they might have been intended. But the flexibility of the software really makes it really easy to once you're familiar with it, figure out ways to adapt it to what exactly you need. So I'm coming up kind of on the end, and we'll take more questions, but this is really the take-home message is that proteomes are more than just abundances. So I talked to you about doing good quantitative proteomics to lay a foundation for any of the functional work that you're doing. I told you about some protein turnover work, using DIA, using Skyline to extract the ion chromatograms. I talked to you just now about post-translational modifications. I didn't get a chance to talk to you about some of my work in protein localization. If you're interested, we can drop some papers. And I'm really excited next to hear more about doing interactomics using DIA from Isa. Big fan of her work, super pumped for that. So that's everything I wanted to cover today. Big thanks to all of my various labs at all of my various stages of training, and I'm happy to take any more questions that you might have and try to rustle up some of those papers if we didn't get to them all. And big thanks to the TAs for covering a lot of the questions. I saw you guys working hard in the chat. Question from Thomas, did you observe if the proteins that had high light to heavy ratio contained more inert amino acid sequences? That is a super cool idea. I like that. I should go back and reanalyze that. We're actually collecting a new data set for that experiment soon. And that's a great idea. Thanks for that. Methionine, cysteine. Yeah, that's interesting. Yeah, I should look for that. What databases do I suggest for histones? I'm not sure what exactly you mean by databases. I don't use any databases. If you mean database search, like search algorithms, I really like Bionic, which unfortunately is proprietary. But Bionic works really great for multiply modified peptide detection. I think Peaks is the other one, which I believe is also proprietary, unfortunately. If anybody has a good free open source suggestion for multiply modified peptides, please let me know. I'm on the hunt for something that would do a good job with multiply modified peptides. Ahmed, is it possible to target peptides that are not prone to modifications for quantification purposes? Of course. So you may have noticed in the IRT calculator, I'll see if I can grab that slide again real quick. In the IRT calculator that we worked on, we specifically tried to get peptides that are not susceptible to modification. All of the N-terminal amino acids are modified here because we perform 2 rounds of chemical derivatization on our histones. So we perform propionylation at the protein level so that all of the N-terminal protein is labeled and all the lysines that are unmodified get modified. We do the trypsin digestion, so everything should end in an arginine, not a lysine, because we've modified all of our lysines. And then finally, we do another round of the propionylation. So now all the N-terminal peptides are modified as well. So these, we tried to get peptides that don't have internal lysines. Of course, a couple of them do, but we tried to focus on peptides that did not have internal lysines so that they wouldn't be modified. Seb, have I ever used DIA for deuterated samples? I have not, but there is no reason that you couldn't use this workflow here with deuterated. You might have a little bit more of an issue with deuteration with the isotopic envelopes overlapping. So I didn't really talk about this, but kind of the idea that people had was that SILAC-DIA wouldn't work because typically the light and the heavy, these are isotopic distributions. And the 2 boxes represent 2 DIA windows, very narrow DIA windows. Ideally, you would see something like this where the 2 peptides, the labeled and the unlabeled peptide, would fall entirely in 2 different windows. But the concern was if you split the isotopic envelope of a particular peptide so that it split across 2 windows, now you were going to get kind of weird chromatogram shapes. So you were either going to get, it looks like half the abundance that it really truly should be, or you're going to get kind of sawtooth shapes as the peptide falls in and out of staggered window resines. So I haven't tried deuterated. You can try and see if you observe something like this or not. Really easy to do kind of those mixture proteome and check the ratio. That's like a pretty good benchmark for that, I think. Raja, it appears that spectral library is a must in analyzing PTMs. That's not true. So that's true for me in this particular instance. But for doing things like phospho, there are excellent phospho repositories out there. And there's several algorithms that you can use to analyze PTMs in DIA data. So Brian Searle, part of the encyclopedia software suite, is an algorithm called Thesaurus. And he used it to analyze phospho DIA data. We'll see if we can get you that paper. It was NatComs, I want to say like 2018 or 2019. NatComs, it was with Judith Vienne and Mike McCoss, but Thesaurus can do that. I know Spectronaut can do at least single PTMs. DIA-NN, Diane, can do PTMs. So yeah, definitely you don't have to have a paired spectral library for multiply modified peptides. I think it's just a problem that's a little bit too hard for DIA right now. So right now, I like continuing to do those DDA spectral libraries. Is fractionation recommended for DIA? What type of gradient is recommended? People have been pushing the gradient length for DIA very short. So there's a lot of papers out there right now doing as short as like 5-minute plasma gradients with DIA or swath. So you can definitely do a short gradient. Fractionation, as far as physical fractionation, is not recommended for DIA, at least by Brian Searle and I, at least. We recommend gas phase fractionation. So you can go back and listen to his lecture, and he touches more on why we do not do physical fractionation for DIA. Thanks for getting my back, Thomas. He's got a good reference for the multiply modified histones. A question about the optimal number of gas phase fractions. We did 6. Yeah, if you go back and you listen to Brian's lecture, I think he did a little bit more elaboration on this. 6 was just chosen because we like to do a range. I can move my slides off probably, huh? We like to do a range. There we go. Of 400 to 1,000 MZ. So when we do 400 to 1,000 MZ, the math works out very nicely to do 100 MZ fractions. And that 100 MZ fraction means you end up with 6. Let's see, what else? Concerning isotopic effects, are there preferences for deuterated or C13 labeling? For me and for those simulations that I showed you, that was all using 13C 15N. So that's like as wide a shift as you can get with SILAC. So as heavy as it gets. When you're going down and becoming and decreasing the space between those isotopic shifts, I suspect you might get more and more concerned with splitting that isotopic envelope like that. But again, I would say try it, empirically try it. I love those Navarro style LFQ bench hybrid proteome style experiments. Do a couple ratios of light to heavy and see if they match what you would expect. I think it's probably the best way to convince yourself if it's working or not. How do you examine the data? Suzanne says, to make sure pulseSILAC forms fall in the same DIA window or in separate windows and not in between when you do not get a jagged profile. So you can do simulations like I showed you. If you look in my GitHub from that paper, I have the script that I use to generate those. You can just kind of type in whatever peptide you want to check and see how it would fall, because you could just calculate that, right? You can calculate what the mass to charge, what the isotope distribution should be theoretically for a peptide sequence, and then calculate what the shift would be if you added the heavy isotope amino acid. So you can do that. I think, again, I think the best way to convince yourself, though, like in theory, that's nice, right? Like theoretically, it's nice to see if that is happening or not. But I think it's even better just empirically to prove to yourself, like, is the ratio not what I expect? If the ratio is not what you expect, you might be seeing some of that isotope splitting affecting your quantification, because it's affecting the transmission of ions through that precursor window in your DIA. But yeah, I use those for everything. I use those benchmarking style hybrid proteome stuff for everything. I love them. Okay, so I think we got all of the questions. Oh, question, when would you use encyclopedia over Skyline? Skyline I always use if I'm doing kind of a targeted analysis. So commonly my workflow these days is I'll do my peak detections and my retention time alignment using encyclopedia. That generates a library. You can straight up generate a blib library using encyclopedia, and then I put that in Skyline, and then I can extract to view and manipulate my chromatograms in Skyline. So if you look at that paper that Brian had mentioned, the MCP paper from 2022 that Brian and I put out, the supplement number 1 is a tutorial that walks through like step by step exactly what we do, integrating encyclopedia with Skyline for our analyses. Do you think DIA could be used for something like thermal proteome profiling? Absolutely. It's all the exact same thing, right? So that's kind of like you're just measuring peptides and then doing some post-processing. So I don't think there's any reason you couldn't do it for thermal proteome profiling. Nice. So we got all the questions.
Well, this is fantastic. Thank you so very much, Lindsay, for the excellent presentation. I am really amazed by what can be accomplished by a combination of instrumentation, experimental design and data analysis all together. You know, this is a really great, great work. I'm really happy I had a chance to hear it.
Yeah, thanks.
So if we don't have any more questions, then it is time for roughly a 15-minute break. So we will be back at 12:45 Eastern, so a quarter before the hour, to listen to Isa's presentation. So please take some time, stretch your legs, and we will be back in 45 minutes. Thank you all. Okay, it is time for us to be back. Welcome back to the Institute on Computation and Statistics for Mass Spectrometry and Proteomics. Before we start, there was a question about the attendees and where people come from. So we have roughly, let's see, roughly 170 participants this week, and between 120 and 160 are in each session. And half of them are from the US and half of them are from everywhere in the world. So we're very excited to actually, it's one of the few good things about the online platform that we can all be together without having to physically travel. So this is very exciting to have such a diverse audience geographically and also hopefully with diverse scientific interests. And so speaking of international participants, so it is now my great pleasure to introduce our speaker who is from far away, who is from Europe. So Isa Bludo did her PhD at ETH in Zurich in the lab of Rudi Abersold and the Institute of Molecular Systems Biology. And Isa did a lot of work on developing software and data analysis strategies for DIA experiments, also focusing on large-scale data sets and large-scale analysis. And some of the applications focused on computational strategies for detecting and quantifying protein complexes. And so since that time, Isa completed her PhD. So she is now a postdoctoral fellow in the lab of Matthias Mann at Max Planck Institute in Munich. And she focuses on analysis of proteoforms, including post-translational modifications and their crosstalk. And I am delighted to welcome Isa. I must say also that Isa was a veteran contributor to the short courses which were offered at ETH for many years. So Isa also has a lot of experience on the educational side. And I haven't heard from Isa for a while, so I'm really excited to hear her talk. Welcome, Isa.
Thank you very much, Olga. I'm extremely delighted to be here. Let me quickly start the presentation. I hope you can see my slides. So I'm super happy to give a presentation today and to share the work that I actually still did in the lab of Rudi Abersold at ETH Zurich. By now, I'm in fact in Munich, the Max Planck Institute for Biochemistry, but the work that I'm talking about today will be focusing on the work that I did in Zurich. And I hope that during this presentation, I can give you some insights and hopefully also some inspiration of how you can expand the concepts of proteomics in general and specifically of DIA to apply them to the analysis of protein complexes and also to assess proteoforms, something that also Lindsay already touched upon in her presentation before. So to introduce the topic of my presentation today, I want to start off with showing this figure which looks at the expansion of molecular diversity in the cell from the genome all the way to the molecular functions in the cell. Because in fact, the genome of the cell only contains in humans roughly 20,000 genes. And this number of genes is then quite dramatically expanded by RNA processing events such as alternative splicing, ending up in tens of thousands of mRNA isoforms, which are then translated to proteins, which we typically analyze by mass spectrometry. So this is actually not the end of diversification, but only the start of more modular and a diverse set of different things that can happen. For example, post-translational modifications, as Lindsay said, can cause phosphorylations or other modifications to the proteins. And proteins can then interact with each other to form protein-protein interactions and also large macromolecular machines, such as the proteasome or ribosome. And as Lindsay already mentioned, the different interactions of the proteins, their localization and their modifications have a very important impact on the protein's function and the different activities they can perform in the cell. So in my presentation, I will now focus mostly on the interactome and how we can extend concepts that you've already heard about for kind of the targeted proteomics to the analysis of protein complexes. And specifically, I will talk about a strategy that we developed in Rudy's lab called SAGA-MS, which we can use to analyze protein complexes on a global scale and across conditions. And finally, I will change gears a bit and talk about how we can actually get a handle on proteoforms in bottom-up proteomics data and investigate how different proteoforms might have an impact on the protein assembly. So to start off, I'd like to start with a very simple representation of, or like a simple introduction of how protein complexes are commonly analyzed in mass spectrometry. And the gold standard to do this is by affinity purification coupled to mass spectrometry, or short APMS. And what you do in this, in APMS, is you select a specific bait protein, and then you fuse an affinity tag to the protein, and then you can lyse the cell and under mild conditions to keep protein complexes intact, and then pull down the bait by the affinity tag, and this will not only give you the bait protein, but it will also pull down all of the interacting proteins. So what this will result in is a very simple interaction network with one central node, which is the bait protein, and you have several interaction partners that are connected to the bait protein via edges. So this is already fairly nice, but one thing that you already can see is the interaction network is still fairly small, and you cannot directly get information about the actual protein complex modules that are there, as for example here indicated by this trimer and the dimer, which are actually separate complexes. So to resolve this, you would actually need to do an orthogonal reciprocal pull down on a second bait, here now indicated with this red protein. And by using the second bait, you can extend, first of all, your interaction network, but you will also already get some insights into modules in the interaction network. If we now, however, want to look at the interactions in an entire cell, then we actually get fairly complex very quickly. So here I'm just showing one example of the massive bioplex effort that has been led by Ed Hudlin et al. And here the goal was actually to get a handle on the entire interactome of the human cell by tagging all human open reading frames. And here you can see the interactome hairball of more than 2,000 baits already. So what I would like to show, what I wanted to show you with this first slide is that affinity purification to APMS actually requires a lot of individual experiments to get a whole interactome. So you can look at APMS as a targeted interactome acquisition strategy. This is similar, for example, compared to proximity labeling, where you fuse the bait protein to an active enzyme that is then labeling proteins that are in close proximity to the bait. And with this, you can get like the surrounding proteins or the microenvironment of a protein in the cell. But both strategies are discrete in a way that the coverage of the complexes that you can investigate with them is directly linked to the number of experiments that you perform. So now the question is if there are any strategies where we can look at the interactome from a more global, untargeted perspective. And the most commonly used approach, and the one that I've also worked with mostly during my PhD, is a strategy called protein co-fractionation mass spectrometry, or co-frac-MS. And here you can take the full native cell and lyse it to keep, again, protein complexes intact. And these protein complexes are then separated by native complex fractionation, for example, by size exclusion chromatography, or short SEC or SEC, or ion exchange chromatography, IEX. The common theme is that you biochemically fractionate the complexes according to some biochemical property, and then you sample a number of fractions across the separation dimension. Each of these sampled fractions is then analyzed separately by bottom-up mass spectrometry, and then what you get are these nice protein intensity chromatograms, where you can see the proteins eluting along the fractionation dimension, and proteins that form a stable complex will nicely co-elute together, as you can see for these two toy complexes here. Now, the benefits of co-fractionation MS compared to other strategies such as APMS is that it is actually independent of genetic engineering or the availability of antibodies, and it can, in theory, allow the parallel detection of hundreds of protein complexes at the same time. And so if we go back to the previous slide and we look at the context of or the scope of complexes we can detect in such experiments, we can see that co-fractionation MS is not discrete, but the coverage of complexes that you can achieve in an experiment is dependent on whether you do some pre-purification, for example, of specific organelles, such as looking only at the mitochondria or the nuclei. But you can also take the full cell, and you will, in theory, cover the entire interactome of the cell in a single experiment. This is, of course, practically not possible at the moment, but this is mainly due to technical limitations. So we cannot cover the entire proteome by our proteomics methods at this stage, but we hope to get there pretty soon. And yeah, so the limitation is not the concept of the data acquisition. On top of co-fractionation MS, there are also other strategies to look at the interactome from a global untargeted perspective. One is, for example, cross-linking of whole cell lysates, which can be done. Another option is to do thermal proteome profiling, or TPP. In TPP, you basically look at protein denaturation curves across increasing temperatures, and what you would expect, that proteins that form a stable protein complex actually show very similar denaturation characteristics in this case. And another alternative is to really look at full proteome covariation, where you can leverage very large MS cohorts and look at which proteins actually co-vary across the entire data set. But in my presentation, I will today mostly focus on co-fractionation.
Isa, there's a question.
Yeah.
How do you handle a large number of baits?
Yep. So this is a very good question, and not necessarily my area of expertise. So I guess so. I don't know exactly what you mean with this question, but you basically have to just acquire data for the baits that you're interested in. So you can, for example, say, okay, you're interested in a certain molecular pathway, and then you select all of the proteins that you know are taking part in that pathway, and you take all of them and analyze them together to get an interactome of this specific pathway or your organelle of interest. But if you have a context or a project like the Bioplex effort, then you have thousands of baits, and this gets very tricky, and you need to have very specific analysis workflows to handle this. I hope this kind of answered the question. So with this, I would now like to move on to analogies between proteomics and interaxiomics data acquisition. So if we look at the MS proteomics side and targeted data acquisition, we have strategies such as selected reaction monitoring or parallel reaction monitoring. And what we do in these strategies is we select some peptides prior to data acquisition, and then we specifically target those peptides during our acquisition mode. This is kind of analogous to the situation in APMS, where we, prior to our experiment, select a specific bait protein that we want to investigate in our data acquisition. So on the untargeted data acquisition side, we have on the proteomics side data independent acquisition DIA that you've talked about already a lot over the past few days. And here the idea is that you extract peptides from an entire M/Z range and co-fragment the precursors. So what you will see is that you actually have a lot of fragment ions co-eluting together, and the situation, and you don't have to select any specific peptides of interest prior to data acquisition. In co-fractionation MS, on the interactomic side, this is actually very similar. So we do the data acquisition for the entire cell together, and you will, in theory, get or like from the conceptual way, you will get protein intensity profiles for all the proteins that you can detect. So if we look at the two strategies on the untargeted data acquisition side, I hope you can appreciate that the data structures actually look very similar. So while on the DIA data set, you have fragment ion intensity profiles across the C18 retention time dimension, and you try to find a peak group of fragment ions that correspond to a single precursor. On the co-frag MS side, you have protein intensity chromatograms along this size exclusion chromatography or other fractionation dimension. And here you try to find proteins that co-elute and form a protein complex signal.
Isabel, sorry,
Yeah.
can I read a question because it might be relevant? Hello,
Yeah.
when using SCC or IEX to perform fractionation of native protein complex, how should we evaluate if the complex dissociate during passing of the SEC or IEX columns? So how do you control that dissociation is not happening during separation?
Yeah, that is a very good question. So generally, so our experience is that this actually doesn't happen too often. So what we mainly have, we look at the protein, like the overall protein content that we have, like after the fractionation, we just check the protein content in each of the fractions, and we should see that there are still a lot of protein in the early fractions that are in the high molecular weight range, and these like general total protein profiles, they actually look very similar across like replicate sec fractionations. And if something went wrong during a sample preparation, then you will actually see that your total protein amount in the fractions shifts. And this is a good indication that, that the protein complexes actually fell apart. Okay. So having said that there are actually these conceptual or like similarities between DIA and co-frag MS data, I now want to get to the analysis strategies between the different approaches. And in discovery proteomics and DIA analysis, we have on one side the spectrum-centric analysis approach, where we start from the data. And here the idea is that we take the spectra, we perform a database search to identify what the data what signal the data corresponds to us coming from. And a similar approach can also be used for the analysis of large-scale interactome data. And here we start from the data. So here in this representation, each of these little dots correspond to an individual protein. And this matrix shows a correlation matrix between the protein abundance profiles across a co-fractionation experiment. And what you might see is that there are actually clusters of highly correlated proteins. And we can find these clusters and group the proteins to complexes. And then we can check in a database or so whether this complex has been annotated before, and these would be the known complexes. And if they haven't been annotated before, we could identify novel complexes. However, we can also look at the interactome data from a hypothesis-driven direction. And here we can first look again at the peptide-centric analysis approach in DIA, where we start from a spectral library, where we have all the information about our peptides of interest and the fragment ions that we want to look for them. And we then go into the data and try to find evidence for these peptides in our data. So we can use or adopt an analogous approach that we call complex-centric for interactome analysis. And here we can take a protein complex database, such as the quorum complex database, and then we can look for evidence of these protein complexes in our data to see if these proteins are actually highly correlated to call this complex detected, or if they are not correlated at all, so this complex would not be detected. So this complex-centric analysis approach is kind of an idea that has been around for some time, and it's not something that we've invented, but it's maybe not always been called like this, but the overall idea has been used in strategies for thermal proteome profiling, where Becher et al. Analyzed protein complex variation across different cell cycle stages, and it has also been used in full proteome covariation studies to look at protein complex conservation across humans and mice. And finally, of course, we have in the Eversalt lab adopted an automated strategy to look at protein complexes and co-frag-MS data by the complex-centric analysis approach. So we have a question here, I quickly check. So which type of software do you use to assess the statistical significance of interaction results? In other words, to check if the enrichment of a certain protein is certain proteins is significant. This is a very good question. So usually what we do is we take a protein complex database, as you can see here, and then we also generate a protein complex decoy database. And I will get to this a few slides down the line. But the idea is that we then compare the number of significant, or like the number of detected signals for the target complexes and compare them to the detected signals for the decoy complexes. And then we can build a scoring and an FDR model for this. So now I've basically showed you these two strategies, and I want to give you a bit more perspective of the limitations and the benefits of the discovery and the hypothesis-driven analysis approach in interactomics. So on one side, we have the discovery approach, which has the huge benefit that we can actually determine novel protein interactions, and we don't require any prior knowledge in form of a complex database or so. But it comes at the downside that you might lose sensitivity and selectivity to detect protein complexes because of the large number of proteins that you look at. So on the complex-centric side, we are in a different situation. So we actually require prior information to start our analysis, and we have the downside that we cannot detect novel complexes. However, we have a significantly boosted sensitivity and selectivity because we can use this kind of target decoy approach to really figure out statistically significant protein complex signals in our data. A third approach that is also under the hypothesis-driven strategy is the network-centric idea. And here, instead of using a protein complex database, we can just use a protein-protein interaction, or PPI, network. And here we don't look at an entire complex module at a time, but really just at single protein-protein interactions to see if we find support of this interaction in our data by a positive correlation across our data set. And then we can enhance this edge in the network, or if we don't find evidence, we reduce the weight of this edge to get an updated interaction network. And this approach has actually the benefit that it requires less strict and less well-defined prior knowledge than the complex-centric approach. And it is very scalable across a large number of conditions because not all of the interaction partners always need to be identified in all samples. So this is a fairly new approach that has recently been published by my former colleague, George Rosenberger, in the SAGA tool. So one thing that I would like to highlight as well is that the world is not completely black and white, and actually there are also hybrid approaches where you can think of actually combining discovery and hypothesis-driven approaches similar to direct DIA strategies in the analysis of DIA, but I will not go into too much detail about this here. So just as a brief summary of the general concepts of interactome data analysis, I'd like to highlight that they are similar to the targeted and untargeted acquisition strategies in proteomics. These also exist in interactomics, and from the data analysis perspective, untargeted interactome screens have a lot of similarities and similar challenges with DIA data analysis, and the hypothesis or complex-centric interactome analysis strategy can be seen analogous to the peptide-centric DIA analysis, and turned out to be a very powerful strategy that has been successfully applied in different contexts. So there's a question: how many fractions do you generally use? If we have multiple replicates, can we mix the fractions based on Western blot? So generally, so I will get to this, but we used mostly like a lot of fractions, so 60 to 80 fractions by a size exclusion chromatography, which is also suggested. Like I would recommend this if you really want to get a good resolution and you want to look at also at complex intermediates and not only at general broader scales. And the mixing of fractions would probably be possible. We have thought about this, but I think this would make the data analysis way more complicated, and I would not recommend this at this stage, I would say. Okay, so having like discussed now some of the general concepts of interactome data analysis and similarities to proteomics in general, I would now like to focus on the complex-centric proteome profiling strategy by Zach Swathames that we developed in the Eberside lab during my PhD. And to start off, I would first like to show you the more regularly used discovery workflow for de novo complex identification in CoFrag-MS data, often also referred to as protein correlation profiling. So this is one of these discovery-based analysis strategies where you start from the protein elution profiles, you perform a pairwise scoring of the correlations between the proteins, and then you can use an elaborate machine learning framework to actually group proteins into modules of co-regulated protein complexes. So while this works very well in theory, there are several challenges associated with this analysis strategy. First of all, the situation is not as easy as shown up here, but we actually look at profiles of thousands of proteins at a time. So in our experiments, we had around 5,000 proteins quantified. And due to the limited peak capacity of the chromatographic fractionation strategies that we have, this has the consequence that we are facing a lot of random coelutions of proteins, and this results in a limited selectivity and sensitivity of these de novo complex identification approaches. I will pretty quickly look at the questions. So one question is, I would think that all your discovery and hypothesis-driven networks analysis on protein-protein interactions will depend, and that should be validated by other assays methods on the type of buffers. And of course, the number of fractions used. Yes, so this is definitely true. So it generally depends a lot on what kind of fractionation strategy you use, what information you can get. I actually have to check what is the stationary phase in the SAG columns, Thomas. I'm sorry. I'm the computational guy in this analysis, but I can send you the reference. So if you check this following paper, you will see all the specifications of the experimental setup that we used. So, oops. How many protein complexes are generally observed in each fraction when you use 60 to 80 fractions? I have to think. So I think this was roughly, so there were quite a lot of complexes. So if we, in a typical experiment, we would find something like around like maybe 400 complexes, and they are not completely homogeneously spread throughout the separation dimension. So on average, I think it was something like, oh, I actually don't, I have to look it up. So maybe it was something like around 10 complexes or so, but might be a bit more. Okay, then there is one question about the machine learning steps for clustering analysis. So basically, what is done in these analysis approaches is that you usually take also a prior information from like high confidence protein complexes annotated in quorum, for example, and these are used as like a ground truth data set for the machine learning, and you try to best recover these known complexes, and then based on this train the machine learning algorithm to group proteins according to the results that you would that you basically optimize for these known complexes in the reference database. Okay, so with this, so what I just said is that we have these random coelutions, and we want to increase the sensitivity and selectivity of our analysis. And one way that you can do this, and what has been done by other labs, is to actually use multiple orthogonal fractionation strategies to get rid, to increase your selectivity. But in the Ebersole lab, we had an alternative idea by coming from the DIA analysis. We thought we could use the complex-centric or targeted approach to analyze our data. And so the strategy that we developed is called complex-centric proteome profiling by 6/4 MS. And from the technical side, we use size exclusion chromatography at high resolution, sampling between 60 to 80 fractions. And we then perform bottom-up proteomics by DIA or source mass spectrometry to get a highly a complete quantitative matrix. And then finally, we play the trick of using the complex-centric analysis strategy, where we use prior information from a complex database to specifically target the complexes in this database in our data. So here I want to again highlight how the complex-centric strategy is an extension of the peptide-centric DIA analysis workflow. So in peptide-centric DIA analysis, we have a spectral library where we have information about specific peptides. Then we go into our highly multiplexed DIA data and extract only the fragment ion chromatograms that we have annotated in our spectral library to do a peak detection to get our peptide signal. In complex-centric proteome profiling, we use prior knowledge from complex databases such as Quorum. We then go into our highly multiplexed SEC profiles and extract only the protein chromatograms of our proteins of interest in the complex that we just query. And then we try to find the protein coelution peak group of the complex in our data. So to perform this analysis, we actually developed the CC Profiler R package. And this is our software for automated complex-centric analysis that allows the parallel and sensitive detection of protein complexes in co-fractionation MS data sets. And as I said, we have a decoy model that can actually perform FDR estimation. So to give you a bit more insight into the workflow of CC Profiler, we take peptide level protein correlation profiling data, as for example, put out by Skyline or Spectronaut, or in our case, we use OpenSWOT, and we import these peptides into CC Profiler. Then we perform some sex-specific filtering. And what I mean with this is that we can, for example, filter out peptides that have a lot of missing values, so that don't have consecutive identifications across the fractionation dimension. And then we can also look at sibling peptide correlations, because usually what we would expect if we have a protein without any proteoforms, then all of the peptides from the same protein should have a similar elution profile and they should correlate well. So we can kick out all proteins, all peptides that don't follow these criteria. And then we perform a protein quantification step where we usually sum up the top 3 proteins to get a protein elution profile. Then we can take protein connectivity information and do the complex-centric signal detection. And for this, we have 3 main steps. The first is the hypothesis generation. So as I said, you can use protein complexes from complex databases, but you can also use interaction networks derived, for example, from APMS studies such as BioPlex or the string database, and use graph partitioning algorithms such as the cluster 1 algorithm or something similar to group proteins into actual complexes. And then in the second step, we generate a decoy database with a matching decoy for each target. And what we require is that the proteins that are in such a decoy complex are not physically interacting or like grouped together in any of the target complexes. So now we go to the coelution signal detection, and here we implemented sliding window approach, where we go over the fractionation data and try to find regions where our target proteins are highly correlating, and we perform a peak detection. Finally, we can do a grid search across a set of parameters, meaning, for example, the width of the sliding window that is used, or the minimum correlation that our proteins need to have in order to call something a peak group. And then we finally can perform an FDR estimation based on our targets and decoys in the database. And with this, we then get a list of complexes detected in our data. So there's one really great question from Ahmed. If we've tried creating protein profiles from peptides apart from summing them up. So we have tried a lot of different strategies, and I will actually get to this later. But if you especially if you have different proteoforms, summing up all peptides or like any number of peptides is not the greatest strategy. Other things that we have tried was averaging, but since we filter out, like in this filtering step that I mentioned here, we are actually filtering out any peptides that don't agree with the other peptides. So overall, we should get very homogeneous peptide profiles. And in these cases, the protein quantification doesn't differ so much whether we take the average or the sum or top n, top 1, whatever you choose. Okay, so as I said, we have implemented our analysis strategy in the CC Profiler R package, and we have recently also published a very detailed step-by-step protocol that walks you through the entire wet lab as well as the computational analysis workflow that needs to be performed to get hopefully some nice results for your data. And now I would like to show you some of the insights that you can actually get by applying the complex-centric workflow to your data. And
there's a question.
Yeah.
So when your output is a continuous area under the curve value for the presence or absence of a peptide, for example, output of skyline, how do you define if the peptide was present or absent in the fraction?
So you...
So Juan, maybe you can ask the question directly.
Yeah.
Hi, yes, sorry, is that the idea, the question came to mind when you were talking about the filtering, that you're checking if, let's say, a peptide was present in consecutive fractions or not. But how would you actually classify if the signal is in there? Like, do you use an intensity threshold for deciding if the area that was integrated is from an actual signal or is just noise being integrated? And if that's the case, how would you define what's the appropriate threshold to use? Not sure if I explained myself very well.
Yeah, no, it's a very good question. So generally what we do is we have we use an FDR threshold on the peptide level. For each fraction separately. So if the peptide drops out of the selected FDR that we choose, then it is we consider this peptide absent from this specific fraction. But this being said, we are actually using a fairly loose peptide level FDR threshold, but rather filter on the protein level. And then we take the SEC information as kind of a better way to look at whether the peptide is confidently there or not, because what we expect are these really smooth elution profiles for the peptides, and that the peptides, as I said, correlate well with each other from the same protein. So if this is not the case and the protein has been, for example, quantified by noise in a lot of fractions, then we will filter it out during this filtering step. But yeah, it's a good question.
This FDR level is based on the identifications of the peptide by the database search, or like from an M profit or something like that?
So this is based on something like an M-profit score. So the combined machine learning model of how you score if a signal is present or not.
Okay, thank you.
But it's not a quantification value, but it's the detection value.
Thank you very much.
Okay, so now I get to the actual data. So the first proof of concept study we performed in a human HEC293 cell line, and here we performed Moritz Heusel, who actually generated all of the data, or most of the data that I'm showing here today. He fractionated the HEC293 cells into 81 fractions, and each of these fractions were analyzed by DIA swath MS on the TripleTOF 5600 using 120-minute gradients, and this led to the consistent quantification of roughly 5,000 proteins after very strict quality filtering. And using now the complex-centric analysis strategy with quorum as the target hypothesis set, we could confidently detect signals for 572 complexes and at a 5% FDR. And we compared this to a manual annotation benchmark where we manually checked all of the complexes annotated in quorum and manually assessed whether we would call them detectable or not. And with our automated CC profiler workflow, we reached a roughly 90% sensitivity at a 5% FDR. So on top of being able to detect holo complexes, we can actually use the data to also derive information about assembly intermediates. And here what you can see in this figure are the protein subunits of the proteasome. So I'd like to take some time to explain this figure. So on the x-axis, you see the SAG fractions from 1 to 81, and they correspond roughly to a molecular weight. And the early SAG fractions are according to high molecular weight fractions because there's a log linear relationship between the molecular weight and the SAG fractions, and we can use a calibration standard to match fragments to molecular weights. And the first fractions correspond to high molecular weights, thus large complexes, whereas the higher SAG fraction numbers correspond to lower molecular weights for smaller complexes or where the monomers would elute. On the y-axis, you see the protein intensity, and the black vertical lines indicate the complex signals that CC profiler detected. And each of the colored profiles that you see here correspond to one of the proteasome assembly members. And what you can see is that the signal on the left here is the highest molecular weight signal that actually where all of the proteins subunits of the proteasome contribute, and this corresponds to the whole 26S proteasome complex. Here to the right, there's a second peak, which only contains diaphragm beta subunits that form the core 20S proteasome. But what you can also see is that CC profiler detected several elution peaks in the lower molecular weight range. And we were now curious to see if these might be disassembly products or if they are actually assembly intermediates or something interesting in the proteasome. So what we did is we actually added known assembly chaperones for the proteasome to our analysis, and they are here indicated in red, orange, and black. And what you can see is that these assembly chaperones nicely coalute together with the alpha and beta subunits, and this gave us confidence that these signals actually correspond to an early and late assembly intermediate of the proteasome. And I think this very nicely highlights the modularity and really the resolution that you can get into molecular details based on this very global data acquisition approach. So finally, we can also evaluate some global proteome assembly characteristics, because as I said, there is a log linear relationship between the SAG fractions and the molecular weight of these fractions. And so for each protein where we know the expected monomeric molecular weight, we can assign fractions to being either monomeric or assembled. So when we look at protein signals, we can figure out if detected peak groups are actually in the assembled or in the monomeric range. And we figured out that 66% of the proteins appear in at least one assembled state. And we could further see that a lot of proteins, namely 27%, have multiple peaks along the SAG dimension. And I think these two results showed very nicely could give a very nice insight into the diversity of assembly states that are present in a single cell, and that proteins might actually be contributing to alternative functions by being part of different complexes. So while this is of course fairly nice to get insights into to proteins in a single state, of course, the question is if we can also look at protein complexes across different conditions. And my former colleague, Moritz Heusel, actually published a very nice study last year where he looked at protein complex rearrangements between HeLa cells in interphase and mitosis. So here I'm just showing a simple example of the chromatin remodeling related BRCA1 complex, and you can see all of the complex members of this BRCA1 complex are eluting here in the early SAG fractions in this experiment in interphase, and you can nicely see that all of them are significantly upregulated in mitosis, which is expected due to the experimental setup. So one kind of bottleneck that we then discovered in this analysis is, as I said, we actually used 120-minute gradients in our initial experiment. And what this results to is approximately 1 week of MS measurement time for just a single replicate and condition. And this made this initial comparison between the cell cycle stages extremely expensive in terms of time and, of course, money. So the question was whether we can actually speed up this analysis in any significant way so that we would make the approach more scalable and actually like a screening technique. So what colleagues of mine did was actually set up a very short EvoSep gradient with only 20 minutes, LC gradients, and with this we can actually analyze one replicate of a SAG experiment in a single day. And what you can see in the bottom graphs here is that this shortening of the gradient to 21 minutes results in a decrease in peptide IDs of 30%, and on the protein level, this accords to 20%. But if we look at protein complexes that can be detected, you actually don't lose a lot of information by reducing the chromatographic gradient. And this makes the entire approach much more feasible for larger studies. So what we did was actually use this very short gradient system to investigate the proteome assembly landscape across macrophage differentiation and stimulation. So we took the THP1 cell line and analyzed 3 replicates of the undifferentiated cells. We then differentiated them into macrophages and then stimulated them with LPS. And for each of these different conditions, we extracted the protein complexes and performed the size exclusion chromatography and acquired the data with the 21-minute SAG-SWOT MS acquisition schemes. So now having this data across a lot of conditions and different replicates, we had to also extend the CC profiler framework. So as before, we have a quantitative peptide matrix and a protein complex annotation as input data. We then import the data, perform a molecular weight calibration, and generate hypotheses. And then we have several pre-processing modules, for example, that include normalization of intensities across conditions and replicates, and an alignment of the profiles between SAG-crons. We then perform our SAG-specific filtering that I mentioned and do protein quantification. Following this initial pre-processing, we can then actually do differential analysis on 3 different levels. The first one is we can look at global assembly state differences. So comparing the assembled mass fraction of a protein. So here we look at a single protein at a time. And again, as I mentioned, we can distinguish the monomeric and the assembled range of the SAG fractions, and then we can compare whether whether there's a shift from the monomeric to the assembled range of the SAG range. The second comparison that we can make is we can look at protein peaks without looking at complexes directly. And here we can see that for this protein, we find 3 distinct peaks along the SAG dimension. For each of these peaks, we can do a separate comparison to see if, in this case, the monomeric protein abundance decreased during differentiation, which would be the case here. And finally, we can perform the complex-centric analysis, where we look at all the proteins of a specific complex query. And here we detect 3 different subcomplexes of this toy example. And we can, for each of these complex instances, we can do a quantitative comparison. So now I would just want to show briefly what kind of biological mechanisms we could detect in this setup of macrophage differentiation. We could see that generally the proteins that underwent any kind of assembly rearrangements are enriched for terms related to extracellular matrix organization and cell adhesion, which makes sense regarding the morphologic change that the cells undergo during this differentiation process. And upon stimulation, we see that pathways that are related to immune response and to virus infections are actually highly enriched, which is confirming that our experiment should actually have worked. And here I just want to show one example of a protein-centric analysis that revealed the WASH C5 protein that moves from a lower molecular weight peak to a higher molecular weight assembly state. And we can see here that a lot of proteins do such a transformation between different of individual peaks. And this is a specifically interesting protein because this protein we wouldn't have picked up by a standard proteomics workflow because it doesn't change its overall abundance. So the abundance stays actually the same across the 3 conditions, but we see this shift from a lower to a higher molecular weight range. If we look at the complex-centric analysis side, we could see that there were a lot of protein complexes that were actually changing upon differentiation, but only a single complex, the IFIT complex, was upregulated upon stimulation. And you can nicely see this here, that the complex is basically absent in the early 2 conditions. And here we have a nice high signal of this complex, which is involved in interferon-induced antiviral response. So before I conclude this part on SAG-SWOT-MS, I would like to highlight one question or answer one question of why we actually want to use DIA for complex-centric proteome profiling. And the simple answer is that we both get more consistent and identification and also quantification. And what you can see here is basically the minimum number of consecutive detections along the SAG dimension that we could see in DIA data compared to DDA data acquired in parallel. And what you can see is that generally we have a larger number of consecutive identifications in DIA compared to DDA. Also, the precision of quantification appears to be much better. So what you can see here on the x-axis is the average Pearson correlation of a peptide with its sibling peptides originating from the same protein. And what you can see is that the average correlation for DIA was much higher than for DDA. And finally, these 2 aspects put together actually resulted in a much better performance in the complex detection for DIA compared to the DDA workflow. However, I have to say that this is all data on the SciEx triple-TOF instruments, so this might look slightly different for different instrument platforms. So to summarize this part of my presentation, I hope that I could convince you that complex-centric proteome profiling is able to detect and quantify hundreds of protein complexes in a single go. And it also enables the investigation of subcomplexes and assembly intermediates, and to look at global assembly characteristics, such as changes in the assembled mass fraction. Furthermore, we can look at complexes differentially and look at rearrangements across conditions. And by using very short gradients, this allows us to measure a single experiment per day, making this much more scalable. And finally, I'd like to say, as mentioned before, that DIA really has some impact on the quality of the data that we can get. I quickly look at the question here. If one does not have the possibility to DIA, is it fine to do 30-minute DDA measurements? So there have a lot of studies have been done with DDA, and I think this is fine. I mean, as I said, there are challenges associated with it, and you might not get like the quality of your chromatograms might not be as nice, and the scoring might suffer from it. So yeah, I would generally recommend DIA if you do DDA. Yeah, I would definitely go for longer gradients than 30 minutes, just to make sure that you actually cover enough proteins. So one other option is that, so I am usually with DIA starting from peptide level information, but you can also use CC profiler beginning from proteins directly. So this might be more optimal for using DDA, because you kind of get rid of problems of single peptides being missing in some samples. And by starting from the proteins directly, this could be easier. Another question by Charlie: can SEC really isolate more than 10,000 kilodalton of protein complex? Yeah, this is a good question. So if we go here, so there is not a high resolution at this really high molecular weights. What we usually see is that in the very early fractions, there are lots of proteins eluting, and they might be membrane associated or like specific to smaller organelles, for example, the lysosomes. And so a really good resolution you will get only in the middle part. So yeah, the high molecular weight range is more tricky. Another question by Thomas: do you think this workflow would be amenable to a middle down proteomics workflow? So this would be fantastic. I would really love to see this combined with middle down proteomics, or even top down. I think this would be fantastic. So I think I don't see why this shouldn't be possible to combine. Only problem might be the sample amount that you need. So you need to make sure that you, I don't know what is currently required for having a good middle down proteomics result. Okay, I think these are the questions. So, so with this, I now like to go back to my introductory slide, and where I was talking about the molecular diversification in the cell. And I will now change gear a bit and focus on how different proteoforms, for example, derived by alternative splicing or post-translational modifications, can have an impact on protein complex formation by first looking at how we can actually measure proteoforms in bottom-up proteomics. So for this, I want to go back to the general principles of bottom-up proteomics and highlight its limitations. So in a standard workflow, we start from proteins, we digest them into smaller MS accessible peptides, and then we perform our mass spec analysis And from this data that we generated, we have a computational workflow first back to the peptides by database search or our DIA analysis, and followed by a protein inference step where we map peptides back to the parental proteins. So while this is fairly simple, if you imagine that each protein would have a unique set of peptides, but in the real world proteome, this is much more complicated. First of all, peptides might be shared between proteins of different genes. And this is a fairly common problem, and it's usually tackled by protein grouping strategies where you, for example, assign a shared peptide to the protein which has more unique peptide evidence. But the situation gets even more complicated if we consider alternative sequences derived, for example, by alternative splicing, and even more when we look at PTMs. And in these cases, we would actually have to perform a proteoform grouping to look at an individual functional species. And usually this is not being done in bottom-up proteomics due to these challenges of having disturbed this direct association between the proteoform and the actually measured peptides. And this is done more typically by top-down. So before we go into more details, I want to briefly define what I mean with a proteoform. So proteoforms, in the way that I define them here, are protein variants that originate from the same gene, but that have a unique amino acid sequence and post-translational modifications. And so as I said, typically we do not look at these proteoforms in bottom-up proteomics. However, now the question is there anything we can do to change this? And the answer is yes, because we can play a trick and leverage information across a large number of samples. So in bottom-up proteomics, we have the benefit that it's fairly easy to generate large data sets, and we can use information from sibling peptides across large data sets to evaluate or to find groups of correlated peptides. So here I want to just show one example where we have two toy conditions, A and B, measured in 6 biological replicates. And what you see are peptide intensities that are all belonging to the same protein, or like the same original gene. And what you see is that the majority of peptides indicated in blue follow the same quantitative pattern, and they increase in abundance in condition B. However, there's a second set of peptides indicated in orange that actually decrease in abundance in condition B. And this can be explained by different strategies, but one thing that you can appreciate already is that if you do a classical bottom-up proteomics workflow and you just do protein quantification here, and you would consider all peptides, this would actually result in a fairly bad quantification based on these orange peptides. So now you can tackle these kind of problematic proteins by filtering out the outlier peptides to improve protein quantification. However, the other approach is to actually assume that these peptides might be of biological relevance, and you can try to find individual peptides or groups of peptides that follow a different pattern and try to see if they actually form an interesting proteoform group. So now I will present one a tool that we developed in the Ebersold and Rist lab that is called correlation-based proteoform assignment, or short COPF. And here the idea is exactly that if you have a single protein that is generated from a gene, then we perform bottom-up proteomic analysis, then all of the peptides in theory should follow the same quantitative trend across a large data set. However, if we have a gene that actually results in different alternatively spliced mRNA isoforms that are then translated to different proteoforms, they can actually no longer be separated in a single bottom-up proteomics experiment. If these two proteoforms are differentially abundant across the conditions that we look at, we can actually figure out groups of co-regulated peptides, which will distinguish the two proteoforms. So now we can actually do this automatically by looking at the pairwise peptide correlations. So here on the left-hand side, if all of the peptides follow the same pattern, they all have a very high correlation and you get a very homogeneous pairwise correlation matrix. However, if you have two alternatively regulated proteoforms, peptides of the same proteoform group will have a high correlation, whereas peptides across the two groups will have a low correlation. So the idea in COPF is that we take these peptide correlations and perform a hierarchical clustering and group the peptides into two clusters based on the correlation distance. And then we can calculate a proteoform score based on the within versus across cluster correlation, and actually also calculate an associated p-value to this proteoform score. And what we will get from this is something that looks like a one-sided pseudo-volcano, where we have the proteoform score on the x-axis and log-negative adjusted p-value on the y-axis. And you will see that proteins with multiple distinct proteoforms end up with a high proteoform score and low p-value. I quickly answer some questions. So does COPF apply to DDA or just DIA? So I have analyzed only DIA data with COPF so far, mainly because it strongly benefits from very good quantitative peptide level information. And this is usually not as good in DDA. But in principle, the method is agnostic to how you acquired your data. Then is the COPF applicable to measure PTM or phosphorylation stoichiometries? In principle, yes. So you can measure PTMs, and you can also try to figure out the stoichiometry or like the occupancies of the PTMs. Although I have to say that I haven't done it myself so far, but it's definitely possible.
Can I add a little bit to that question? Because yes, as it is the case with many PTMs, unless you perform enrichment, you will usually not see them. And that changes, well, the complete peptide mixture that you're analyzing, and you wouldn't be able to see the picture of the whole protein anymore without disturbances due to the enrichment. Will it still be possible to do the analysis like this?
Yes, so I will get to this maybe in a few slides. So we actually did perform a targeted extraction of phosphopeptides in our data. So maybe I postpone the answering of this question to in a few slides.
Thank you.
Yeah, I will probably get to the rest of the questions a bit later, just to get through the main points first, and then we see how much is still open in terms of questions. So just very briefly, I think I don't want to spend too much time on this, but we benchmarked the COPF strategy based on an in silico generated ground truth data set. Basically, the reason why we had to generate something in silico is because there is no good ground truth data set of where we know exactly what proteoforms exist. But in our in silico benchmark set, we could see that we get a reasonable true positive rate at low false positive rates. But what is even more important is that we actually have a working scoring model with well-calibrated and overall conservative FDR models, where you can see the adjusted p-value that COPF estimates against the empirical FDR. You see that we are generally following the diagonal and are slightly on the conservative side, just to show you that the overall method should be sound. So with this, I'd just like to do a very brief disclaimer, because there's a very distinct difference between proteoforms that can be detected in top-down proteomics, because in top-down, we can actually fully characterize a protein with unique amino acid sequence and localized PTMs. In bottom-up proteomics and the COPF approach that I'm presenting here, we can only look at proteoform groups, and these are sets of co-varying peptides in our data. that are differentially abundant across our samples. But the key here is the differentially abundant across our samples, because what this means is that, per definition, all of the proteoform groups that we detect are kind of biologically interesting because they have to be differentially regulated in the data set that we're looking at. And this is, of course, very interesting because we get just a few way fewer hits than with top-down, but the ones we get are potentially of high interest. So with this, I now like to get back to the SAG-SWOT-MS data because our idea was not actually to do this on a multi-condition data set, but to look at proteoforms and how they might be assembly specific. So the overall idea here is that the different SAG fractions basically work similar to having a lot of different conditions. And if we look at peptide intensities across the SAG fractions, then we might figure out whether proteoforms are specifically integrating into a complex or not. So here in this toy example, I again show all the peptides as lines, and they are all peptides from the same protein. And what you see that there are two orange peptides that are only present in the low molecular weight peak. And this might be very interesting because then we could say that these two peptides are actually unique to the monomeric form of the protein, and they are absent in the complex bound form. So we applied this strategy of using the Kopf approach for SAG-SWOT-MS data by using the data set that I briefly introduced before, where we looked at SAG-SWOT-MS data of interphase and mitosis cells. And here again, I can say that we could detect both proteoforms that would be due to a difference in assembly state, or they could be, as here, actually a difference in assembly and in condition, or like cell cycle specific. So applying the Kopf strategy to the cell cycle data set, we could actually detect 317 significant proteins. So these are proteins that were called by Kopf to have multiple different proteoforms. And this was at a 10% FDR and a proteoform score cutoff of 0.1. And what you can see is when we performed an enrichment analysis of the UniProt keywords, then phosphoprotein and alternative splicing came up. So this makes sense in context of finding proteoforms. And also the other terms were largely related to cell cycle regulation, which makes a lot of sense in the biological context. When we further looked at the classification of those proteins, we could assign roughly 100 proteins to be assembly specific, so they differed across the SAG dimension, and like around 120 or so were cell cycle specific, and they were actually differentially abundant across the two cell cycle stages. So now I would like to show you some examples of the data. So one is like just a proof of concept example of the nuclear pore complex protein NUP9896. And this is a very interesting one because it actually is coming from a single gene and single mRNA, NUP98, and the precursor protein is autoproteolytically cleaved in the middle to result in two proteoforms, NUP98 and NUP96, which basically operate as completely separate proteins. If we look at the peptide profiles of all the peptides that map to this gene, you will see that you get very distinct sets. So here, all of the lines correspond to peptides that map to this gene, and the colors of the peptides are determined by the groups that Kopf assigned. So what you can see that the yellow and the blue proteoform groups actually have very distinct elution patterns along the SAG profile. If we now want to see whether these two proteoform groups determined by Kopf actually correspond to these two proteoforms. We mapped all of the peptides to the protein sequence, and you can see that all of the blue peptides map to the N-terminal region of the protein, whereas all of the orange peptides map to the C-terminal region. And the autoproteolytic cleavage site actually lies exactly in the middle. So this was for us a very nice proof of concept. And what you could actually see is if we do the complex analysis in this data, you will see that the orange proteoform, so the NUP96, is actually integrating in the NUP107-160 subcomplex, whereas the NUP98 protein is not contributing to this complex, which is something that has been known before, but we could do this in a completely data-driven way in our data. So the second example I'd like to show is about an example of proteolytic processing of the proteasome subunit beta type 7 and during proteasome assembly. So what is happening during proteasome assembly? You first assemble all the alpha subunits to a ring. You then assemble the beta subunits on top. And one of the last subunits that enters is the beta 7 subunit. So once the subunit is integrated, it actually needs to be proteolytically cleaved for the full 20S core proteasome to assemble. And when we look at the beta 7 protein and all of its peptides in our data, you see that all of the peptides follow the same pattern, apart from two peptides that Kopf segregated as a separate proteoform group. And these two peptides only contribute to this low molecular weight signal over here, which you can see magnified in log scale over here. So what we know is, as I showed already in previous slides, this high molecular weight signal corresponds to the full 26S proteasome. This is the 20S core proteasome, and this is actually an assembly intermediate over here. So when we looked at the peptide location on the protein sequence, we could see that the two blue peptides actually mapped to the N-terminal region, and the second blue peptide was spanning the known proteolytic cleavage site where the beta subunits propeptide is cleaved off for the assembly to take place. So what we then wondered is whether we can detect this non-tryptic peptide maybe in our actual 20S proteasome. So what we did is we actually went to Skyline and leveraged this like the beautiful feature of DIA that we can go back and re-extract information that we didn't think of before. So we took the non-tryptic peptide spanning this propeptide cleavage site and performed a targeted extraction. And you can see that we've detected a very nice peak this is now C18 retention time dimension. But if we look at the different fractions here just shown in the Skyline representation as different samples, you see a nice elution peak in both interphase and mitosis. And if we aggregate the signal, we actually see that this non-tryptic peptide nicely elutes together with the rest of the 20S core proteasome. So one thing that I would also like to get back to is something that was already asked in the questions, but that already came up in our analysis here, is that phosphoproteins came up as a main term in that was enriched in our proteins that we call to have multiple proteoforms. And and the idea here, or our question, was whether we can detect these phosphopeptides in our data. And our idea was that we could use evidence by absence. So what you could imagine is that if you have a protein and you can see all of its peptides in one condition, and then there is a phosphorylation happening, then the non-modified peptides would actually disappear. So here you see the two orange peptides are highly abundant, but upon phosphorylation of both of these peptides, they might disappear from your analysis. So we thought, okay, this might be a way, even though we didn't do a phospho analysis itself, to figure out such cases. And indeed, we found several very interesting cases. So here is an example of the ataxin 2-like protein. And you can see, again, all of the peptides for this protein here in these two plots. And you'll see that there were 4 peptides indicated in blue that are highly abundant in interphase, but they are actually decreasing significantly in abundance in mitosis, whereas all of the other orange proteins actually increase in abundance during mitosis. So now we mapped all of the peptides again against the protein sequence, and we saw that they actually distributed widely across the sequence. But then our idea was maybe these are phosphopeptides. So what we did is take a data set, an external data set that was investigating phosphorylation in the cell cycle. And we figured out that there were actually 5 regulated phosphosites that are cell cycle specific. So that I indicated them as kind of these lollipops here on the sequence. And what we saw is that 4 of these known cell cycle regulated phosphosites are actually directly mapping onto our 4 peptides of the second proteoform group. So now we were wondering, if we go back into Skyline, if we could actually extract these phosphopeptides if we look for them. So here, just show one of the phosphopeptide examples. So we did a targeted extraction, and we were super happy to see that we could actually detect a very nice signal for this phosphopeptide in our data. And here again, you can see that there is a nice elution of this peptide across the samples in mitosis. And this is actually in 10 to the 6 range here. And if we look at the interface, we don't really see any elution, like large peak. So this is again sec dimension now, and we are at peak areas in 10 to the 3. So with this, I can conclude that we could actually detect 3 of the 4 phosphopeptides that we hypothesized. And for 2 of them, we even get a very beautiful coelution signal with the other orange peptides here shown in brown and in red. So with this, I'm almost done. I just want to briefly give a quick outlook that COPF is not only applicable to co-fractionation MS data, but it can also be used across different conditions. So we applied it to different tissues of the BXD mouse panel. And so here we measured 8 BXD mice and took 5 different tissues and looked at whether we can find any tissue-specific proteoforms. And overall in this data, we could see 63 significant proteoforms, proteins with proteoforms, and almost all of them we could confirm to be tissue-specific based on a condition-aware ANOVA analysis. So I just want to show one quick example where of the cipher protein, and here again you see all of the peptides of the proteins and the 2 proteoforms that COPF detected in orange and blue. And what you can see is that, first of all, the protein is very muscle specific. It's only expressed in heart and quadriceps tissue. And the orange peptides are only observed in the heart. If we now map these peptides back to the protein sequence, we could see they actually span a known alternative sequence region. So what we did is, again, look at what is the alternative splice variant of the protein in this region. And we performed a targeted extraction in Skyline and could actually detect peptides in this alternative sequence region. And these peptides were actually then skeletal muscle specific. So what we could do is basically figure out, based on our data, that there are 2 distinct isoforms of the cipher protein that are heart or skeletal muscle specific, something that has already been known before, but that we again could see in a completely data-driven way from our data. So with this, I get to the summary. Basically, I hope I could show you that correlation-based proteoform analysis by COPF can use covariation patterns to assign proteoform groups in bottom-up proteomics data, and it can detect proteoforms derived by alternative splicing, proteolytic cleavage, or indirectly even by PTMs. And in combination with SAGA, this actually allows for the first time to systematically assess relationships between proteoform groups and assembly and complex assembly. And with this, I get to the main take-home messages from my presentation. I introduced you to the general concepts of targeted data analysis and how they can be transformed from proteomics to interactomics. And I hope I could show you that SAGA is a very great strategy in combination with complex-centric analysis to get consistent detection and quantification of complexes, even across different conditions. And finally, I showed you the correlation-based Kopf analysis, which can allow you to look at proteoform groups in bottom-up proteomics and investigate proteoform-specific assembly characteristics. So just for you to know what you can do with this, so all of what I just talked about is available as R package in the CC profiler framework. So also all the Kopf analysis steps are available in CC profiler, and you can find it on GitHub. And there's not only the package itself, but there's also a complete walkthrough the general steps of the standard complex-centric analysis and even of the DIA analysis with OpenSWOT. And you can find more details in this protocol paper. And for the Kopf analysis, I've also set up a Git repository, which you can find on the bioRxiv paper. So with this, I'd like to thank all of you for your attention and for the many questions, which I hope we'll get to now. But I would also like to thank everyone that was involved in these studies that I presented, especially Rudy and my colleague, former colleague Moritz Heusel, who, as I said, generated most of this data, and the other collaborators on all of these projects. And yeah, now I try to get to the questions.
Thank you so much, Isa.
Okay, can you quantify a single protein in different fractions or complexes? Usually one would expect different overall protein expression in different protein complexes. Okay, yeah, so we can detect a single protein, definitely can detect a single protein in multiple different complexes. This is basically what the entire strategy of this proteoform specific assembly boils down to, that we have the opportunity to actually see that proteins elute multiple times in the SAG dimension. And we observed this for more than like around 30% or so of the proteins that actually have multiple elution peaks along the SAG dimension. Then question by Raja: do you identify occupancies without enrichment step for phosphopeptide occupancy? Yeah. So yeah, as I think I could show you that we could see phosphopeptides also without the enrichment step in our data, but we actually did perform also a systematic phosphosite analysis, but this was not so spectacular. But overall, this works in the data, especially if you have enough fractions, then you can actually extract quite some PTM information from the data, as you could see also with the targeted skyline extraction, where then you would hope to find both the modified and also the unmodified peptide. Another question is, how do you process such large number of samples, all fractions together, or replicates together? Yeah, this is a good question. So I don't know what you mean with process. So in terms of the wet lab protocol, we actually do one SAG experiment at a time, but each sample is handled separately. We now have like a 96-12 format where we can actually do this in parallel. For the data analysis, I actually, for the Kopf analysis, use a combined information from multiple replicates to get this information. So I actually do an aggregation step before I call the proteoforms. Yeah, so this is how we do this. Then one question, can COPF take any relative quantitative data, such as TMT? I would actually have to try. So in general, it should work. So if you have, for example, 0,1 normalized data, I don't see why it shouldn't work. So the general idea is that you look at the correlation, and this should be agnostic to the absolute values. So if you didn't mess around with the ratios between samples, this should be fine. But I haven't tried it, and I'm not a TMT expert. Then one other question, how does the SEG-SWOT approach compare to genomics-driven proteoform discovery? I guess the SEG-SWOT filters for functional differences of proteoforms. Yeah, so I guess with the genomics-driven approaches, you're referring to RNA sequencing approaches for alternative splicing, for example. And what we saw is that we can, of course, only see a fraction of what can be discovered by genomic approaches. So, for example, you can detect hundreds or like thousands of alternative splice variants, which we cannot detect by the COPF approach. But as you also correctly said, what we can detect are actually functionally relevant proteoforms that show a difference between conditions. So we are blind to any kind of proteoforms that are just present in the cell but don't have an impact, but we could see the ones that are actually different across our data set. Yeah, one question, does it have to be SWOT-MS for DIA? No, definitely not. So you can use any DIA setup that you would like to. So this is completely platform independent. And could you quantify the phosphorylation in protein complex? Yes, so I mean, in general, we could, I mean, this is what I just showed in the slides. So this is, so I actually don't know which complex this corresponds to, but what I know is that this is not the monomeric form of the protein. So we could see that this protein elutes in a higher molecular weight than you would expect for the monomer, but we don't know exactly for this example what the interaction partners are. But in general, you can see the complex information for these phosphopeptides in our data. Okay, then one other question, can COPF be used to analyze regular DIA data, not the co-fractionation? Yes, so this is exactly, I think I answered in the last data with the mouse tissues. But what is important here is to say that we really need a lot of variation. So in this data set, we have a very diverse set of conditions, and we expect almost all of our proteins to change in abundance. And this is kind of very crucial to the strategy, because this correlation-based approach only works if you have differences. And if you don't have any differences, you will get non-relevant correlation information. And then what was the reproducibility of your peptide detections along experiments? So I assume that you're talking about proteoform detection. So this is actually fairly high. So as I said, I did this actually for the different replicates combined, but I also performed initial analyses on each of our replicates and conditions separately. And for the assembly-specific proteoforms that were non-condition specific, we could see a very high reproducibility of the calls across the different samples. Then have you tried with IEC with SWOT-MS, and if so, is there a significant difference between SAG methodologies? I haven't personally tried, so there are a lot of different strategies that you could use. I know that other people in the Ebersold lab have also used native page separations, and this worked extremely well. for other sets of complexes that would, for example, also be larger and potentially membrane associated. I have not tried it with IEX, but from the methodology, like from the data analysis point of view, it works for all kinds of separation techniques, although the idea of using the molecular weight as an additional factor only works for separation techniques that actually involve the molecular weight of the complexes. Okay, and I think there's one final question. Can you use the SAG-DIA coupled with other bottom-up or top-down MS to determine the stoichiometry within selected protein complexes? And can you convert the abundance factors from mass spec analysis for one selected protein into copy numbers in the complex? Okay, so in terms of stoichiometry, this is actually a very cool question. And we have looked at this in context of the septin complex, which is present both as like in multiple forms, and they have multiple exchangeable subunits. And what we could nicely see is that the exchangeable subunits are at a much lower relative abundance compared to the constitutive subunits. And we could even calculate these stoichiometries, but unfortunately our data is maybe a little bit too sparse to get like a full-blown picture of how well this works. So I would say, so CC Profiler actually reports stoichiometries, but you should look at them with caution, I would say. In terms of copy numbers, I haven't done it, but in theory, it could be possible if you calibrate it right. And another question, in complex protein discovery, can you summarize the weak points of SAG-SWOT and the bioinformatic workflow exposed here? So the weak points, I would say, are that you definitely need prior information, as I said. So this means that if you use a non-model organism, for example, you're depending on information that might be from homologous mapping or things like this, and you have just very little information. And then I would say that the complex-centric analysis workflow doesn't necessarily work very well. The other thing is that you need quite some material to like from the experimental side to perform like a full-blown SAG-SWOT MS experiment. And I mean, I think it works already fairly nicely, and we could do it with tissue samples, for example, as well. But overall, you need quite some amount. So it's still some way to go, for example, to get to clinical samples. And one maybe other point from the bioinformatics side, what did I want to say? The weak points is that so this was now the weak points of the bioinformatics workflow is that you need a sufficient resolution to actually be able to derive signals with this. So if you, for example, have noisy signals, this gets very, very challenging, and you will need sufficiently high quality data for the analysis to overall work. And of course, SAG-SWOT MS in itself is a strategy for kind of stable complexes. So if you want to, for example, look at transient differences, like very small differences between perturbations where you have small, like some minor rearrangements in complexes that are not super stable, I would not recommend to use SAG-SWOT MS because you will see the rearrangements on the big scale. So if you have a massive rearrangement of complexes, as for example in the cell cycle or during differentiation processes, then this is a great strategy to use. But if you're more interested in like minor perturbations, I would recommend something else.
Well, Isa, thank you so very much. Thank you for the presentation. And let me also thank Lindsay and Isa at the same time for spending time with us today and sharing their research. Before everybody leaves, so there has been a little bit of a discussion in the background about where people come from and how far they had to travel online to join us today. So those of you who are here, I am launching a poll right now, if you don't mind, so that we can take a look. If you're still here, let's see who is coming from where, and as soon as we have enough responses, I will share with the entire group so that we have a sense of where our participants come from. And we have some compliments in the chat. Thank you, Lindsay and Isa, for the talks, and also compliments in the Q&A box. Thank you everyone. So let's see, we have 6 out of 79 people answered. Let's give it a couple more minutes if some others would like to join. Okay, 68 answered, a couple more. Yeah, don't be shy. It's all anonymous anyways, we don't know where everybody is coming from. Okay, 9 more people. Sweden, yes, we have some chat messages as well. Okay, 70 out of 79. Let me end the poll and see if I can share results with everybody. Yeah, so we have a lot of people, roughly 40 people come from North America, a few from West Coast, I guess it was very early, 20 from Western Europe, but we have also people from Eastern Europe and Asia. And I know that as far as registrants in the entire program, we have quite a few people from Australia as well, and quite a few people from Latin America. So thank you very much for joining us from far away. Maybe we will do this poll once again later, maybe next week, just to see who actually stays with us for that long. But it was a pleasure to hear from Lindsay and Isa today. Thank you for your contributions, and thank you, Juan and Diana, for TAing so effectively. Really appreciate that. And tomorrow, and actually for the next 3 days, our plan is to dive deep into R. So Friday and Saturday, we will have R for beginners. So if you have never ever touched R, this is the time for you. We hope to take it slowly and interactively so that by the end of the 2 days, you will be able at least to start some analysis on your own. On the other hand, if you do have some expertise in R already, please join us on Monday with Kylie. Kylie has written a lot of very high performance implementations in R and is a very accomplished educator. And I'm sure you will learn new things, even if you have done work in R already. And then we will go on into MS Stats and related topics. And at the end, we will talk also about massive and reproducible research. And the very last day, Alexei Nesvitsky will talk about FragPipe workflows as well. So we will be going into label-based methods. And I'm happy to see Lindsay dog. I was waiting for Lindsay's dog slides all morning.
I know I didn't have them in there today, but she obviously had to make an appearance.
That's right.
Always.
Well, thank you very much. I see other comments saying it was the best session yet. Really wonderful. And see you tomorrow if possible. Thank you, everybody.
Thank you.